Good morning, one and all present here. It's Saturday morning, and we all have gathered here for an enriching session on biosimilars. I, Ms. Anupama Tomar, welcome all the participants for the national webinar Biofacet organized by the Department of Microbiology and Biotechnology at Patkar-Varde College. I would now request our principal sir, Dr. Shrikant Sawant, to introduce our college.
Thank you. Good morning to you all. We are assembled here for the national webinar on Biofacet Biosimilars. Department of Microbiology and Biotechnology presents RUSA-sponsored national webinar Biofacet Biosimilar. Chikitsak Samoa's Sir Sitaram and Lady Shantabai Patkar College of Arts and Science and Patkar-Varde College of Commerce and Economics welcome you on this occasion. Our college was founded by Chikitsak Samoa, an association of like-minded individuals who were keenly interested in education. The college was named after Sir Sitaram Patkar, a distinguished judge of Bombay High Court who was knighted after his retirement, and his wife, Lady Shantabai Patkar. Sir Sitaram was deeply interested in furthering the cause of women's education and was the Vice Chancellor of SNDT University for many years. The College of Arts and Commerce, Arts and Science started functioning from the academic year 1964-65 and the Commerce section from the academic year 1980-81. To provide career-based education, the college started degree-level self-finance courses from the year 2000-2001. Since then, the college has constantly upgraded the courses offered and the infrastructure required for these courses to provide students with best possible learning opportunities. In keeping up the spirit of learning and growing through criticism, we have set up high standards, working continuously towards advanced goals. Many of our alumni hold very good positions in various fields. Patkar-Varde College is a place of pride among us the various education institutions in the western suburbs of Mumbai. Our college is re-accredited by NAC in its third cycle with A+ grade, securing 3.53 CGPA. Autonomy was conferred on the college from the academic year 2019-20. We are ISO 9001-2015 certified, and we are recipient of Best College Award by the University of Mumbai in the year 2016-17. We are DBT Star Scheme and RUSA 2.0 awardee. India's Education Excellence Award also awarded to us in 2018 Berkshire Media LLC USA. Education World India ranked our college 45th in India and 10th in Maharashtra amongst the private autonomous colleges. It's my proud privilege to welcome our eminent resource persons today on this webinar. Dr. Dilip Pawar, Director and Head Clinical Development and Medical Affairs. We have Dr. Jayachandran Ramlingan with us here, who is Head of QC Biosimilars, Serum Institute of India Limited, Pune. Dr. Sanjeev Gupta, Advanced Biotech Biosimilars Research and Development, IPCA Laboratories Limited, Mumbai, and Dr. A. Ramkishan, Deputy Drug Controller of India, CDSCO East Zone, Kolkata. We have a team of eminent resource person for us. On this occasion, I also welcome our Honorable President, Sri Kishorji Rangnekar, Dr. Kiran Mangaonkar, Vice President, Dr. Gurunath Pandit, Joint Secretary, and all the office bearers of Chikitsak Samoa. Dr. Malakarkar, CEO, and the program is organized by Department of Microbiology and Department of Biotech. Today, we have researchers, academicians, students, I'm quite sure that this national webinar will give us insight and there will be nice deliberation on this occasion. I wish you all the best for this national webinar. Thank you. Thank you very much.
Thank you, sir. I extend a warm welcome to the guest speakers of the day, Dr. Dilip Pawar, Director and Head, Clinical Development and Medical Affairs, Dr. Jayachandran Ramlingam, Head QC Biosimilars, Serum Institute of India, Dr. Sanjeev Gupta, Senior General Manager and Head IPCA Laboratories, Dr. A. Ramkishan, Deputy Drug Controller of India, CDSCO East Zone, Dr. G.V. Pandit, Joint Secretary, CEO Dr. Mala Kharkar, Principal Sir Dr. Shrikant Sawant, Chief Coordinator Ms. Ruchita Rane, department staff members and all our dear participants. Department of Microbiology and Biotechnology has been organizing Biofacet every year. Started in the academic year 2012-13, Biofacet is a stride taken by the department to provide a platform to scholars, teachers, and experts from various fields of science and technology for academic interactions, elevation, and enrichment. It has also taken initiatives in helping students in extending their minds to diverse fields available in biological sciences. And since 2018-19, we are successfully organizing national conference. This year, we organized a national webinar on plant health management and an international webinar on vaccine development process. This would have not been possible without your support and participation. Thank you very much, dear participants, for encouraging us to organize Biofacet on diverse topics. We are sure that this webinar would certainly complement to your learning objectives. The topic of today's webinar is biosimilars. Biosimilars are not new drugs, but rather they are copies of biologic drugs that have been used to treat many diseases and medical conditions. To understand biosimilars, we have four eminent speakers who will be joining in each session and would share their knowledgeable experience in the field of biosimilars. Before we begin with the webinar, I request all the participants to be kindly patient if any technical problems arise during the webinar. We will try to resolve all the technical glitches. Kindly post your questions in the chat box. It will be answered at the end of each session. Now, I would request Ms. Sridevi Devadiga to introduce the first speaker of the day. Thank you.
Yes, thank you, Gamana. It is my pleasure and I am honored to introduce our first eminent speaker of the day, okay, Dr. Dilip Ramdas Pawar, a delegate and dedicated AVP and Head of Global Clinical Development, including APAC and developing countries, with 20 years of experience in clinical research, medical affairs, and regulatory in pharmaceutical industry. He has an excellent experience in early clinical development program for research molecules on oncology, cardiovascular, diseases, arthritis, diabetes, gastrointestinal diseases, autoimmune disorders, and pain management. He is a member of Indian Pharmacological Society India, is a vice president of Indian Society of Clinical Pharmacology and Therapeutics, Mumbai, and life member of American Society for Clinical Pharmacology and Experimental Therapeutics, USA. In 2020, where the entire world was suffering, Sir was in the frontier and won several of the awards, which include Indian Medical Association Corona Warrior Award in the year 2020, Times of India Corona Warrior Award on Doctor's Day, Indian Red Cross Society Corona Warrior Award, Mumbai Municipal Corporation PDBA Hospital, Corona Warrior Award, Corona Yodha Award by MLA Vidhan Parishad, Government of Maharashtra, even one award from India Today, Top Mumbai Doctor Award for Corona Warrior in August 2020. Sir is an honorary army doctor treating Indian military and Mumbai police. His list of achievements goes on and on. It would be very difficult for me to list them all today. But Sir is currently working with Unichem Laboratories as an associate VP, involved in medical affairs, clinical development, and pharmacovigilance. He is also responsible for global medical affairs, global clinical development programs involving supervising preclinical programs. Is in charge of global safety programs and pharmacovigilance, and has many, many other roles under his belt, tied to a very long, successful career in his field, decorated with achievements and awards. I finally request Sir to take over the session, and thank you.
Thank you so much, Sri Devi, and I hope I am audible. And thanks to the management of Patkar College and especially Pandit sir. Pandit sir is even my sir, so I have very high regards and respect for Pandit sir. Uh, and thanks for the kind introduction. As we are all aware, for last 1 year, the whole world was suffering with the COVID, and though my specialty is different actually, so I'm more into clinical development, clinical as a physician. I have shifted my career path to develop a lot of new molecules, new products, bringing them to India, and my more focused area is oncology. So I'm super specialized in oncology, and that's why more studies we are doing in oncology area, and that's why the role of biologics and biosimilar. Currently, I'm also working on some of the early development and phase 1 studies. But when the world was facing with the COVID, we thought it is the right time that when your expertise are required, so it doesn't matter what specialized area you are or which faculty you belong to. Actually, there was no compulsion for us, but we still came forward, and from day one we joined. In fact, some of the treatment protocol we were involved in developing that, and if you see, the India was in the forefront in developing this kind of program. So whether it was debate whether hydroxychloroquine can be used as prophylaxis or not, and the whole world said no, we said yes, we should do that, and that's why we could save so many uh, lives of so many doctors and frontline workers. In fact, I remember when WHO also said that initially that wearing mask is limited to doctors and everybody should not wear. In fact, from the early days, we promoted, and we had meeting with the government of Maharashtra as well, and we said that wearing mask should be made mandatory, compulsory. So because we understand, and that's what where the research comes. And why I'm telling you all this, because you as a student, you as a faculty, should always think out of the box. And it is not necessary when we say out of the box thinking means you have to do something large or something big, but even simple things can be reinvented. One simple example I want to give, that most of you must have got information that during COVID, steam inhalation is very important. You must have read a lot of research papers on that. But probably you must have not read who are the authors. In fact, I was the first author in all this publication. So it was our research we studied. So in theory, we came to know that this is very useful. And some of the countries like Japanese and some of the population were already using this. So with that, why not actually check and understand? And then we did the clinical trial. We found out it is so much useful, and that's why we did clinical trials in two major hospitals of Mumbai, and we published. Based on that, I was interviewed by USA channel, by Italian channel, some European channels, and in fact, I asked when we had having interview with the Italian doctors. So I told them that this is a research, but why don't you validate that? And they also did the clinical trial, and they also found that steam is really helping. So the point what I'm trying to drive, that you have to be innovative in life. When you get opportunity, take that opportunity. Don't be afraid of taking that opportunity. Don't be afraid of falling down. Don't be afraid that you might go wrong. There are possibility that you might go wrong, but doesn't matter. And this is how the research happens. And this is how we were able to help so many patients, not only in the India but throughout the world. So thanks for kind introduction. So I like to share my presentation. So can you see the presentation? Excuse me, can anybody confirm? Hello? Can
Yes, sir, we
anybody?
can see your screen.
So you can see the presentation?
Yes, sir. You can, yes, sir.
Okay.
Yes.
So thanks for that. So now the today's topic, and it's very interesting and very important in today's scenario, that what are biosimilars, and what we are going to discuss is the current perspective. And I have heard that there are some eminent speakers after this, so I won't devote much of my time in explaining those expertise which requires a lot of QA and those data, because that is very important in biosimilars. And being a clinician, I like to highlight overview over the biosimilars and what are the concerns usually a clinician, a doctor, when he treats a patient, has in this mind. So thank you all for joining this, and once again thanks for giving this opportunity. So let me start my presentation with the famous words spoken by Mother Teresa. We can do no great things, only small things with great love. And in fact, in today's scenario, this is very, very important. We have seen last 1 year, everybody was struggling with the pandemic and everyone was affected. And this was the first time that doesn't matter we are rich or poor, doesn't matter what area you belong to, what specialty you belong to, everybody was affected. And that's why it's very important that don't think of doing great thing, even the small help what each individual gave. Maybe somebody was standing on the road and providing water, somebody was providing food to the building watchman, somebody was providing fuel to the migrants, or the doctors, staff, police, everybody was involved. And this is how the science plays a lot of important role. And that's what I'm trying to highlight earlier also. Don't think of doing one great thing. When you try to do even smaller things, and when you collate, that becomes a big research, that becomes a big activity. So try to pay attention on smaller things of life, the basic things in life, and from there you can really achieve great heights. So with that, let me start my presentation. So we all know the biologicals are referred as biopharmaceuticals or therapeutic biologicals. The main difference between the chemical entities and biologicals, because these are produced by living entities. The other major difference, it is quite complicated technology. We'll see some of the overview, and during the later part of the day, the other eminent speaker will touch upon how exactly it is sophisticated. In today's area and today's scenario, the vaccines, as all of you are aware, and India being the front line in producing this kind of vaccine. So now you can understand the importance of this science. In fact, when the whole world was debating, but the Indian pharma people, the Indian pharmaceuticals, the government, everybody joined hands together, and they paid attention. Eventually, if we want to develop this vaccine, don't think of India, think of the world. And this is how today you will be happy to know that COVID vaccine, the Covaxin, and the Covishield, not only in India we have close to more than 3 crore people we have vaccinated. But along with that, we have provided this vaccine to close to now 42 countries. So just imagine the power and the dedication and the expertise what we have in the country. So never underestimate yourself. There was a time when we used to import vaccines, and there was a clip going around on WhatsApp messages that earlier we have taken vaccines from Canada, and today we are providing Canada to the most developed countries. And India has done as a country a phenomenal job, and each one of you have played an important role. So again, coming to the point, always pay attention to the basics in life. So whether it is your life, or whether it is science, or whether it is medical science. So when a patient comes to me, we always try to see what are the basic things in the patient, what are the basic symptoms he has. Even in this COVID period, we know even if simple patient may have mild fever, but don't underestimate. So if you pay attention to basics in life, you will definitely excel and not only in life but also in your research. So more or more we are going to focus on the research. As I was talking about the small molecules, like simple example like aspirin, which have so low molecular weight, but as the molecule, right, that's why we say them as small molecules. All the chemical molecules, what we say them as a small molecule, and all the biologicals we call them large molecule. So if you see how the complexity increases, see the molecular weight, how it increases. Say insulin, then erythropoietin, and nowadays we have a lot of monoclonal antibodies, and see the molecular weights are in more than 150,000 daltons. And because this has become so complex, and thereby the whole concept of biologics is very difficult. And then when you want to have a biosimilar, now what is a biosimilar? Bio is life, and similar means to mimic. So further it becomes very difficult. Why? Because you have to actually match that. And anything which is derived from a live organism, it is very, very difficult to have 100% replica of that. So what is biosimilar? As we have discussed, bio and similar means something which is derived from living organism. It has to be similar, and that's why the biosimilar. So it is successor to a biologic medicine, okay, which is already there in the market. And typically, of course, we have patents. So till that time, you can't make them. But when the patent is over, and then when the generic company wants to make it, so they have to really match. So it becomes very, very difficult. We'll see in the future slides how difficult it is. So it is not a simple generics, but due to, as we saw, the size, structure, then the whole lot of different manufacturing process, because it requires so many different steps, and that's why it becomes difficult. But then it's very important that biosimilar has to be there in the market, because lot of diseases, these are the targeted therapies, especially in oncology and a lot of immune disorders. This plays a very important role, and thereby that's why you need to have biosimilars in the world. And thereby there are a lot of guidelines which has come up, so that we can actually have the same efficacy, but more important, it has to be safe as also. Then there are different names given to biosimilars. People call them fall on biologics or similar biological medicinal products, and different terminologies has been given. Now, though it was a layman understanding what is biosimilar, but various regulatory authorities, so EMA has been forefront in this, that is European Medical Agency, and various the top regulatory agency, whether it is US FDA or PMD of Japan, and of course India CDSCO. So each one has given their own definition. So more or less, if you see, they are quite similar. But everywhere, if you see, the most emphasis is always given on safety, efficacy, that is potency, and the quality or purity. So any biological should at least match these three. We will see how complicated it becomes. Why safety? Because as we have seen, it is derived from live organism. So even if it has good efficacy, and we're not going to have safety, for example, because it is derived from live, so there can be chances of lot of immune reactions, anaphylaxis, antibody production. So it becomes a challenge. Potency, that is efficacy. So one has to pay attention whether it is efficacious, and then it has to match, at least, if not superior, but at least it has to match with the innovator. And finally, the purity, or what we call as the characterization. And I think so later in the days they will talk more about characterization, becomes that becomes very critical, very important, especially for biosimilars. So in all this definition, these are major pillars of these definitions, and without this, a biosimilar will not be certified. When we compare the generics, so as I said, generics normally called small molecules, they are very standard, traditional small molecule, and they are relatively simple and easy to make. Even by back engineering is something easy to make, and you can really make exact replica. But biosimilars, what happen? They are not copies of the originator. Okay, they can be at the best similar, because manufacturing a biopharmaceutical, as I said, it is a complex, and even a small change, if you do, even in that biosimilar, the whole properties changes. So you need to try to make as similar as possible, and that's why the different guidelines says it has to be close to them. Sometimes even if you use the same vector, but still the vector which the innovator has taken was a different, and this live organism which you have taken as a vector as a different, so the properties will anyway change. Like we know, even if two twins are born, they may look similar, but there will be so many different things both of them they will have. So what are the differences between small molecules, or we say the generics, and the biosimilar? Now try to understand. The biosimilars, as I said, it's a large molecule, and you have seen that example. The weights are into hundred thousands. Whereas small molecule, they are simple, smaller in size, and easy to make. The source, as I said, biosimilars always derive from a biological, that is a live source, and that's why the challenge is. Whereas small molecule is always from chemical synthesis, this is machine. So you set the parameters and you can manufacture it. Very important is the physicochemical characterization. Now this becomes mainstay of any biosimilars, and more emphasis given into this complex analytical characterization, because without characterization it is very difficult to go to the next step. We'll see that pyramid. So this is the heart, and this is one of the biggest complex part. Whereas in small molecules, there are standard analytics, your standard methods, and you can easily replicate that. The other things which are more required for biosimilars and which are usually not required for small molecules are basically your bioassays, in vitro, in vivo. Then preclinical toxicity becomes very important. For all biosimilars, you'll have to do preclinical toxicity. For small molecules, usually it is not required because it is available in the literature. They have all the data, and that data can be referred. But you can ask me that why then the toxicity which innovator has done, so why we have to repeat that? Yeah, I agree that you don't have to repeat the entire gamut, but yes, you have to repeat many of these studies. Why? Because your source is different, your live organism is different, your mammalian cells can be different, your bacterial cells can be different, and when these are different, then the product which you are going to get will be definitely different. So there can be various changes in that. And then very important in the air, you have to purify that. And again, when you purify, again the steps, the methods are different. So we are not sure whether the final product what you're getting, how purified it is, whether it still carries some of the toxic things. And that's why the preclinical studies becomes important. And finally, definitely you have to prove, you have to do your clinical part, that is PKPD studies, and finally you have to do clinical trials with the reference product. But here only advantage, the clinical trials are shortened compared to the small molecules. But small molecules, usually what happens, only bioequivalence study is more than sufficient. And clinical trial, like either doing phase 3 study, is not required for generic molecules. But in biosimilars, you have to do even clinical trials. Of course, it is shortened, but then you'll have to do your phase 1 and phase 3 and then confirm the efficacy and safety. And this is what I was trying to explain. Size and complexity makes huge difference. So aspirin, like it has 21 simple atoms, then human growth hormones are around 3000, and your large molecules or large biologicals like IgG has 25,000, and some of the monoclonal antibodies what you have seen may have 100,000 or 150,000. So it differs. It is simple like that. Your simple small molecule is like a bicycle, then your growth hormone is like a car, and then whatever you have, the large molecules or your maps, they are like fighter jets. So this is how complexity, and this is how it differs. Size does matter. Why? Because this biopharmaceutical relates to the protein substance itself. And protein, as I said, there are heterogeneity. Then there are proteins are mixtures of families of the molecular species, and thereby it can have a lot of impurities and contaminants. And that's why again and again trying to emphasize the process and product related impurities cannot be kept completely by analytical means. And that's why characterization of this molecule becomes very, very important. And thus, what are the challenges faced by biosimilar? As I said, manufacturing process, though even if you know the innovator has done this way, but you have to replicate everything and each step is different. Extrapolation of different indications again is a challenge. But now regulators allow that, and I'll cover later part in my slide. The other big challenge what you have is immunogenicity. As I was talking about, because these are derived from your live organisms, and thereby whenever you derive from live organism, the chances of immunogenicity will be always there. Like vaccines, the current scenario, the chance of immunity will be there. Now again, if you see the current COVID vaccine also, if we are giving now, though we are giving the same dose to all the people, but what we have seen, the generation of antibodies will differ from each patient. We have seen some patient getting the titer of antibodies which are in hundreds and 200s, and some patient we have seen no antibody titers generation. So it is such a difficult science to understand. From zero, it can vary to any amount. Now you can ask, then suppose if this vaccine is giving zero antibody titers, and then whether then the vaccine is be useful? Yes, it will be useful, because some people can get titers very fast, some people will get titers later, and even if they get titers later, but still the memory will be there in the cells. So there's a different science altogether. But what I'm trying to explain, the immunity becomes very important, and each patient will respond differently. Then we need to have a lot of post-marketing surveillance data. Pharmacovigilance becomes very important. And some part I'm going to cover the real world evidence, which more and more regulators are giving emphasis on. Next week, I have one more similar webinar where I'm going to talk more about, you all can attend that, more about the real world evidence. And only there we again cover biosimilars, but then more and more regulators want you have to generate the real world evidence, because doing clinical trials for every indication is not possible. And then the other things which becomes issues, that as I said, the clinicians always face challenges, the patients always face challenges, and finally the cost, time, and harmonization about these biosimilars. These are all challenges faced by biosimilars which we try to overcome. Now this I was trying to explain you earlier, the regulatory requirements. And what was one, most of the regulatory requirements are more or less similar. And I was talking about the, if you see the pyramid, the base of the pyramid, these are very, very important. The physico-chemical characterization, the biological characterization are very important, because it has to match, because there is so much of differences in your production, so much of differences in your purification, fermentation. So each process is different. There are more than around 5,000 steps involved, and each step you have to characterize, each step has to be the same. And once you are able to characterize your molecule, and then you assign, okay, now this molecule we have finally characterized, and this is not going to change, and that becomes your reference, which is almost matching to a innovator. Then you enter the preclinical stage. As I said, again, you have to repeat many of the preclinical studies, if not all, but you have to prove that in your preclinical it is absolutely safe. And once you do all, then the clinical part, where my role begins, is actually a smaller part of your pyramid, but the very critical part. And thereby you have to prove your PKPD data, and finally clinical. So usually phase 1 we do where we confirm the PKPD data, and then there is no requirement typically like phase 2, and then you have to jump into phase 3. So once you have PKPD data, then you can eventually start your phase 3. And again, the sample size will depend on what indication you're looking at. And once you are able to give sufficient data on your phase 3, that is multicentric trial, like we got approval for trastuzumab in USA. So we had done study in around 500 patients. So this how the phase 3 will help. And then finally, if your clinical data you are able to prove at least it is similar, then you get approval from the regulatory. and then this data is accepted by many countries, and then the part of real world evidence which I was talking about starts, and thereby, because approval is given for one particular indication, and as I said, a molecule can be used for different indication, and it is not possible for us to do clinical trial for all the indication. Why? One, it will consume a lot of time. Second, now though we have covered all the characters, so it is safe, so it is assumed that then and this molecule will have similar therapeutic role in different indication, and thereby the extrapolation is allowed by most of the regulators. But we don't have data on the clinical trial for that indication, and that's why the real world evidence, so this becomes very important. Now this very briefly I will touch upon, because in the later part of the webinar there are experts who deal with this, so I can give you an overview, but then experts will explain you in detail wherever you want. But to understand the basic, as I said, it is a very highly complex. Now you have your reference product, a genetic code, and then this genetic code from your cell bank you take, and then you have a DNA vector. So you have a specific live organism, a vector you choose, and in this vector then you incorporate your genetic code, and then this you take to either bacterial or mammalian cell lines. Okay, so it can be your E. coli, or it can be your fungal, or it can be a mammalian cell, anything, and then you incorporate this, and then there's a multiplication happens, and when the multiplication happens, so there will be a huge number of cells which will be available, and that's why the complex thing. Now out of this so many hundreds and thousands of cells, you have to pick up the best cell which matches, and then that this best cell again you try to multiply, and that becomes your reference standard, and then the whole process of fermentation, and then purification, and then formulation, so all this, these are the standard parts. But now you try to understand, like each fermentation will be a different process itself, and then after the fermentation you have to take out the cells, and you have to keep your molecule, and then the process of purification starts, and then you have to formulate. So it overall, all this becomes a very complex process. Okay, so even if we say we are getting the same drug, but if you see there are so many parts, so even if a biosimilar uses the same human gene as the innovator, but you see at all the steps it is so different. So whether it is your mammalian cell, bacterial cell produces protein, it will be different. Fermentation process will be different. After fermentation, whatever you are getting, the purified part, that will be different. And because your live cells are different, so that means the even the produce what you are getting will have some kind of influence of your live organism, and thereby whenever you have this formulation, so we are not sure how much reaction or how much immunity it will provide, and thereby you have to have the whole lot of studies including to study the antibodies preparation. So biologics are unique as I said, but complexity of the product and its manufacturing process are different. So thus, though you can see that they look similar, but as I said, there will be whole lot of different available. So try to try to match as similar as product, but you won't be able to produce exact same replica. Okay, so thus biosimilar, as I said, it is a copy version of an already biologic product which is available. But what is important, that you should have similarity in physicochemical characteristics. Again and again, paying attention that your physicochemical characteristics are very, very important, and these are the very crucial part. And once you are able to do that, then the clinical part starts, that the preclinical part forms, where we establish the safety and the efficacy. So not all follow-on available are biosimilar. As I said, biosimilar only applies when you are able to show that your product is similar in head-to-head quality, non-clinical and clinical studies. Okay, so try to understand that everybody will not be able to match, especially the physicochemical characteristics, as it forms a major important part. And if you are not able to match that, so it is very difficult for you to go into the next part of preclinical and clinical. So biological products which are not shown to be similar to a reference product should not be called a biosimilar. So try to understand. Then if you are not able to match that, then again your regulators may ask you to conduct whole lot of different studies. So if you want to match a biosimilar, you have to match all the properties. So if anybody can guess, it's very difficult. So in this, there is one original Charlie Chaplin, and the remaining are just duplicates. In fact, there was one. I remember one story. There was one competition that who can exactly play role of a Charlie Chaplin. Okay, and that competition was open for all, and that competition, so people participated, and there were judges and all, and then they gave the rating. Okay, this was the guy who was first, was as close as Charlie Chaplin, then second, then third, and the funny part was, so in that competition, even the original Charlie Chaplin has taken part, which nobody was aware. And you know what was his rank? He was said you were third in the competition. So sometimes what happen, even original doesn't look like original, and that's why biosimilar is a game where you want to match to the biosimilar. So even if you are good, you may be better, but then if the only innovator has done this, then you have to match the innovator. So that's the funny part. So it is something like that. What you see is a iceberg. So what you see is the tip of the iceberg. The whole lot of process is a huge, the pharmacokinetics, the physico-chemical characterization, the analyticals are deep science. And once you have the product, it is very difficult to understand how much it is required to bring that molecule. So it is so much complex. So what you see is only the tip of the iceberg. The other important thing which I was trying to highlight is immunogenicity. As I said, it is derived from live bacteria. So anything which comes from the live, so whether it's your product or vaccine or anything, it has the potential to have immunogenic reaction. Now, what are the challenges in this? The main challenge is if your immunity is very high, that even it can lead to product elimination or inactivation. Now, suppose if your bodies are going to generate good number of antibodies, so that can kill your product itself, or it can become inactive. So you have to do this immunity studies separately. The clinical consequence of immunity may be benign, or it can be up to severe. As I said, it can be absolutely inactive. And the most threat is about allergy and anaphylaxis. Again, as I said, even in vaccine, what we are seeing, most of the patient may not get, but certain people do have anaphylaxis reaction. Because again, why the chances are high? Because it is from the live source or live organisms. The key issues for immunity for these protein drugs, it can be host related or it can be product related. So host related, we know, as each human being is different. As I said, when we are giving vaccine, some people are getting antibodies which the titers are above 100, 200, some are not getting, some are getting very midway. So again, it is all host related, because how each one's body response or immune response, it will be completely dependent on that host. And again, it's a complex science. So this is one of the major challenges. The other challenge is concurrent therapy, because many patients will be on concurrent therapy. As I said, we have a lot of biosimilars for oncology. So there can be immunosuppression already there in the patient. So whether this is going to suppress that, or there can be antibody reaction. So very permutation combination will be there. Whether there will be activated immune system due to infection, ethnic sensitivity, because as I said, again humans are different. Again, within the world, and there are different countries and different ethnicity are there. So each one will have his own genetic pattern. And even if you see in our country, again we have different ethnicity. So south is little different than the north, susceptibility difference. As we all know, like for diabetes, the south population is more susceptible to diabetes because of again genetics. So even within the country, there can be so much of host related problems. Product related, we have seen most of them, because your each product will be different, the characterization are different, the impurities will be different, because you are deriving from different source, different bacteria, different mammalian cells, then your impurities will be different. Why? Because your purification process is different, formulations are different. So at each level, there will be differences. And as I said, there are more than 5,000 steps involved, and each step can be a potential challenge, and thereby biosimilar becomes more and more difficult. So human reactions can lead to various things. So a biotechnology therapeutic will definitely give you antibody response we have seen earlier, and it can vary. What it can also lead to, as I said, because it is derived from live source, anaphylaxis can be there, infusion reaction can be there, cross reactivity, as I said, because it is a antigen, it is a live organisms or derived from live organism you are incorporating. So it can create a cross reactivity, and thereby it can even lead to inactivation, as I showed in the earlier slide. As we are aware, there can be adverse side effects because of this, altered pharmacokinetics there, and we have to establish that because of this there is no loss of efficacy, because if your cross reactivity is there by endogenous protein and it is interacting with your drug, the efficacy can be lost. Okay, so this whole lot of reactions can happen. So what I'm trying to highlight, so it is a first of all complex thing, manufacturing is complex, characterization is complex, and even if you have successfully made the product, your antibody response can be different. And even if the product is good in terms of all characteristics, but still the host response, the patient response, what can be so much different. So overall you have to pay attention to each part, and thereby large clinical trials for pharmacology can identify the immunity reaction, because as I said, in clinical trials you have very limited number of patients, like what we did with trastuzumab, we had around 500 patients. But then most of the regulators expect that we should have a robust pharmacovigilance system. So I had the pharmacovigilance program globally, so we are aware about it. And not only that, we have to also have a real world evidence data collection, and this we have to keep on comparing, and this is a live comparison. So you cannot just say it's a historical data available and thereby it will not have, no, because as I said, your each batch manufacturing, because each batch will be using a live organism for production. So in fact, even after some time, you have to recharacterize, because what happens? Your first batch, suppose is derived from one of the bacterial cell lines, and suppose if you are going manufacturing your 100th batch, so now the 100th batch, whether it matches with your first batch, that itself has to be seen. And then if there are some changes, so you have to recharacterize. And thereby, because of the various challenges, whole of the world has brought their own biosimilar guidelines. So EMA was the first one to do that. Then WHO has bring out, so that they want to see that more and more biosimilars should be there in the market, because the patient needs, the human population needs it, but it has to be globally acceptable. And thereby they came upon these guidelines similar to biotherapeutic products. And what they emphasize, that it has to be quality, which is your characterization, safety, as I said, safety very important, thereby you need a lot of preclinical studies, and efficacy. And this again, there are three main points which most of the regulatory guidelines or regulator expect you to do. So most of the regulators pay emphasis on, based on the scientific rationale, due to complexity of these biologicals. Again, what they emphasize, your quality, safety, and efficacy. And based on that, you design your preclinical plan and your clinical development plan. Okay. The other thing what regulators have applied, which is good, as I said, there's extrapolation of other indication across indication. Suppose the product is approved, say for example, oncology indication, and the same product is used, for example, your rheumatoid arthritis, then it's not important that you have to do trial for all the indication. So that extrapolation is required, and this was I was trying to explain, that real world evidence comes into picture. So when they approved this product, but since you have derived from a different source, then you have to actually have a good pharmacovigilance program, and you should have a real world evidence data collection, and then this data has to be submitted to the regulators. Why? Because two things: whether your product is giving the similar efficacy, and whether your product is safe. So thereby real world evidence becomes very important. So again, similarly, what WHO has emphasized, on the concept of similarity, the sequential development, you have to play emphasis on the class, product specific, that is a characterization, and finally the preclinical data. And once this is done, then again, as I showed earlier, the pyramid, you have to do the PKPD trial and head-to-head clinical trials. And usually you do phase 1 and then you can jump to phase 3. And once you are able to establish at least non-inferiority trials, then approval is given for all the indication, and thereby they expect you to have a robust pharmacovigilance plan and collect the real world evidence. Following this, CDSU was also laid down, that is our own regulators. They have laid down their guidelines in 2012, and this is in place again, but it is more or less simple and it follows the same like global side. So we have different regulatory agencies like IBSC, RCGM, GCA, AC. This is all related to biosimilars, like genetic engineering appraisal committee is there. Then CDSU, that is our own regulator, who give you permission for the large clinical trials. So whenever you want to apply, all this committee, you need to get approval from them, and then finally clinical trials, and then finally you get approval within the country. Then for, as I said, monoclonal antibodies, so similar guidelines have come by EMA or EU and US. So they have the similar guidelines what they have set up, a stepwise approach for demonstrating the biosimilarity. So EMA guidelines for biosimilar antibodies, as I said, whenever a biosimilar is there, there will be antibodies generation and each will be specific. So they expect you to have preclinical data in vitro, in vivo, and finally the clinical, where you have to do comparative clinical trials between biosimilar candidate and reference antibodies. And then you have human PKPD data. This is slightly different, and this slightly becomes difficult because you have to really define, and there is lack of appropriate define your PD endpoints. Thereby, you have to define this and then initiate the study. So you should have your data on antibodies as well. So are clinical trials required? As I said in my pyramid, if you see, yes, it is definitely required. And it has to be adequately powered, randomized, parallel group, comparative clinical trials mostly has to be done because you have to compare with the innovator. Okay. So you have to follow all the standards, but only the sample size may be required is not like that of innovator, and you have to have adequately powered. Okay. So relevant model population, you have to see that sensitive endpoints you have to include in your study, and you should at least have normal equivalence trial. So normally not non-inferiority trials are accepted. At least you should prove that your product is as effective as this molecule in terms of efficacy and of course safety. So as we have now gone through the whole lot of process, and this summarizing all, as I said, if you remember that pyramid which you have shown earlier, so your physicochemical characterization is very important, that is quality. So for generic, as we see, it is quite easy and we all know it's standard and it's compared with the reference product. But for new biologic, it is an individual quality assessment, and when you are biosimilar, as I said, you have to actually match with the biosimilar. And as I said, sometimes, as I showed you the Charlie Chaplin story, the original can be the the copy might be better than the original, but no, you have to match because they have a whole lot of data with that reference, and it's a very comprehensive comparison with the reference product. So that's a big difference. Preclinical usually in generic we don't do, but full clinical is required for if you new biologic. But as I said, in biosimilar, not all, but you have to have a abbreviated program. You have to have minimal preclinical program for that. And clinical, as I said, for generic it's only bioequivalence study because characterization everything is very standard. For new biologic, as I said, you have to do a complete clinical trial program which includes phase 1, 2, 3 and risk management plan. Here biosimilar has little advantage that if you get a good phase 1 data, then you don't have to go through the phase 2, you directly jump with a multicentric trial with only one indication. But if you are a new biologic, you have to do clinical trial for all the indication, and that's where the big difference comes. So here in biosimilar, you do study with one indication, and then you extrapolate the data and you get approval with the other indication. That approval is given because you have to submit your risk management plan you have to have your pharmacovigilance plan. You will need to have your real world evidence. Based on that, these approvals are given. So based on this entire presentation, the concept of biosimilar is built on these main 5 pillars. As I said again, the physiochemical characters becomes very important. So you should have analytical similarity, preclinical similarity, clinical data should be similar, your efficacy as well as safety. You should have a proper quality system. I think so today later one of the eminent speaker will talk on quality because it's very very important. As I said, characterizing of these molecules are very important in biosimilar. And lastly, as I said, once you get the approval, the pharmacovigilance plan has to be robust because you are going to collect data in different indications. And finally, what we get is real world evidence. And for biosimilar, real world evidence are playing a major important role. So this is how, as I said, showed even a small change in the large molecule. If you see, the post-transit modification may impact your antibody activity. This was I was trying to tell. Even a small antigen is modified, it will impact your antibody activity. Okay, so it will have your altered affinity, it will have your altered pharmacokinetics, it will have altered antibody generation, it will have inactivity, inefficacy, so many things. So small change in your antigen, so antigen antibody reaction will be different. And even a small change, even a single even a single small change can make a big difference. Okay, so even if you remove say one of the amino acids from your this part, so even the characteristic of by removing one amino acid chain anywhere, it will result making a big difference. And that's why this is a very complex process and we have to be very very meticulous in that. And finally, when we do all these things, finally when you have the product, we have not just made that product just to match. Why we have made? So that it should be made available to the patients. And that's why the clinician role, people like me have huge dilemma. What is the dilemma? Of course, why we want biosimilars? Because it is to be affordable. And thereby regulator said, okay, you have do it one indication, and with other indication we can extrapolate the data so that you don't lose a lot of time and the drug is made available to large human population. And that's why it's affordable. Similarity, whether it is 100% similar, as I said, it cannot be 100% similar, but it can be close to in terms of CMC data, in terms of quality, preclinical, clinical. Then you need to have evidence. So based on our clinical trial, we need to PKPD data, we need to create evidence. And the trial data, as I said, you need to have phase 1 and then phase 3 trial data. And more importantly, you should have data on the immunogenicity because these are all derived from live organisms, and thus each data, each time point is important. And then finally, as I said, you should have a robust pharmacovigilance plan and robust program where you can have real world evidence. So when the product is made available in different countries, if you can collect the data, because we have seen so many differences are there in human population, and then whether your product is still maintaining the same efficacy and same safety need to be established. So this is how a biosimilar is. So you can think both are apple, but both are different. So with that, I end my presentation. Thank you so much. So if there are any questions, I'll be very happy to take it. Yeah, organizer, shall I stop the screen now?
Yes, sir, you can, sir. If there are any questions, please post it in the chat box. So we have one question from our faculty, which says, what are the potential benefits of biosimilar to treat cancer?
Yeah, as I said, biosimilar are playing main role nowadays in treatment of cancer. If you see, There are a lot of biologic molecules available, and there are a lot of monoclonal antibodies available which are used in targeted therapy. And as I said, in cancer, what happens? The disease may be spread across, or it may be limited to some organism. But when we have a chemotherapy, in chemotherapy, what happens? It is not targeted. So what happens? The drug is goes into your systemic circulation, and it will kill the bad cells, but in that process it will also kill the good cells, and thereby when you see there are a lot of side effects and complication in this cancer patient. But when we have specific targeted therapies, now this as a biosimilar, this will go and target to those cancer cells. So thereby you have two advantages. First, it kills the unwanted cells, and other, it will spare your normal cells, thereby less side effects, and you get more and more efficacy. So these days, as I said, biosimilars and monoclonal antibodies are playing a huge role in uh, oncology indication, as well as a lot of immunological disorders like rheumatoid arthritis or psoriasis. So it indeed has a very important role in oncology patients.
Okay, what is the most potential danger that you think is connected to biosimilars?
Yeah. So if you remember some of the slides which I showed, the worst potential danger can be anaphylaxis, because if your purification process is not correct, or if there are some toxins which are available, as I said, it is a very complex process, and the purification has to be very robust, very good, and thereby you should have a very good clinical and preclinical development plan, and thereby they play a lot of important role in preclinical. But if you ask what is the severe risk, yes, anaphylaxis can be severe risk. The patient will have a lot of complication immediate, and some patient can die because you are taking something from a live organism. But this is rare. It is not common. It is very rare, but then it is a potential risk. And what are the other risks? As I said, though you have made the molecule, if your immunity reaction is very high, then there is a potential danger that your product can be inactive, or it will be less effective, and thereby you expect results from the patient, but you don't get the desired results. But these are not severe. Eventually, in your real world evidence data, you will come to know. Okay.
Yes, sir. We have questions in the chat box. There's one question which says, does biosimilar have the same route of administration of the dosages?
Yes, yes. So biosimilar, you have to match everything same. It has to, because if you change the route, if you change anything, then you have, it's like a then different route. So you have a different drug delivery system. So you have to take a different path, and then more clinical data will be required. But when you want a biosimilar, it has to be the same path they have to do.
What makes an ideal biosimilar?
Yeah. So there is nothing ideal in this world. So biosimilar will remain biosimilar. But as I said, the more and more you are close to that, uh, as I showed one slide, the pyramid, the more and more you are close to the physico-chemical properties, okay, then more and more you will be near to that biosimilar. But you will not be able to 100% match, for the simple reason, because even in biosimilar, or even in the innovator, for example, the first batch which they say they have derived from some one set of E. coli, take for example. Now that E. coli will not be there after 10 years, no? You will have a different bacterial E. coli, correct? Now again from this different bacterial E. coli, so in technically speaking, if you see, and that's why I say you think out of box. So technically, even the batch which I am taking is after 5 years, that is also a biosimilar. So it is a biosimilar for innovator also. So and that's why innovator also has to do recharacterization. And then I am a biosimilar company. So I am trying to first match the innovator. Now innovator has changed that batch number 10th has changed a little bit. So even I will get change because I have to keep on matching. And the worst part, or maybe funny part, so my product may be superior, but we don't want superior. As I said, Charlie Chaplin may be very good, but he was third in the rank. So I don't know how to prove superior, but I have to match that what they have done. Or if I feel I'm superior, I'm good, then I have to, as I said, then you are not a biosimilar. Then you say my path is different, I'm more better, and then you do the entire clinical development program. You have that option open. So you decide what path you have. But biosimilar, very difficult to have ideal even for the innovator.
So is it possible that if somebody claims that both the products are 100% biosimilar, how true would that be? Or how far from truth is that particular sentence?
No, that so if it is 100%, they will not call biosimilar. So when the very fact they say, okay, I'm biosimilar, that means you are similar, because your characterization will differ. It will not be 100% matching.
Yes. Identical and similar, two different things. Yes, we have more question which says, what is the cost associated with developing of a biosimilar?
What?
What is the cost associated with developing a biosimilar?
Yeah, it is a huge cost involved. But compared to biologics, the biosimilar is lesser cost involved, but the cost is huge. Each will go in crores, and the whole setup and everything is costly. But as I said, think out of box. You have the idea. It is not as costly. The systems and the things are not as costly as your small molecules, because it requires a smaller setup. So you can have a small setup, and then we want to scale up. So there are two kinds. One, you do your research in a smaller setup, and then there are a lot of labs available where we can promote. So you can do your research in a very less cost, and when you want to expand or scale up, then you can go to a bigger setup. So think out
And
of box. So as a student, as I said, never think that you have to achieve something big at one go. Do small steps. And there are a lot of, if you see, when we travel abroad, we see a lot of companies of 4 or 5 people. So they have idea and they outsource this small activity. And once they see, now probably this looks like a better, and then they get seed funding and all, and then they outsource a lot of this activity. Why I know? Because there are a lot of small labs in India, and there are a lot of these entrepreneurs abroad. So what they will do, they will outsource this small activity to Indian R&D companies. And when they see that, okay, now they can see a success or a product, and then they get seed funding, and then they tie up with a bigger company. So nowadays, a lot of permutation combinations are available.
what is the time associated? Is it more than a generic or a normal small molecule drug, or is it more in average?
Definitely, it is more than a small generic molecule, because these are standards. You have all the analytical methods, everything here, right from selecting of your bacteria, cultivation, fermentation, cleaning, characterization. It takes definitely longer time.
Okay, any more questions? Any more questions,
Any,
participants, please?
any more clinical questions if you have? I think so. Theories are too many, and I think so. Your other speakers will talk more on characterization and quality and this and what are challenges they face. Now, as I said, one part is to reach there, make the molecule, and then in clinical parts also. Over the even clinical, we face a lot of challenges, right from your PKPD studies, clinical trials. A lot of challenges are there. So if there are no questions, or I can log out, and if you get any more questions, you can always send me email, and I will be happy to reply that.
Is there any more questions? Okay, we have a question. Okay, for cirrhosis, will completely cure it or will it need
For
doses?
what? For what?
For cirrhosis,
Psoriasis.
Psoriasis.
Psoriasis. See, psoriasis is again a immune disorder. Okay, and again, psoriasis is not completely curable, but yes, nowadays we have good drugs, so it is completely treatable. Okay, so it depends on, again, psoriasis which type you have, and it can be very well treated and very well controlled. So again, it's a immunological disorder, so that can be flare-ups depending on the various environmental conditions. It depends on a lot of genetic makeup of a person. It depends on a lot of stress factors, so this can aggravate. And thereby, in psoriasis, lifestyle modification becomes very important. And of course, it is treatable condition. Nowadays we have good drugs, and it is very well treated. So treatment is not only with the biologics, it is also, there are some good small molecules which are available, but more and more people are referring to biologics. Like one of the product which I had worked was the itolizumab, which was recently also used in COVID. So it has a very good results in psoriasis.
Is there any more questions? I guess I have no more questions. Yes, sir. I'll go ahead and thank you for the session. I wish to wish. I used to wish my express. I would wish to express my gratitude to you and for providing encouragement at biosimilars and interchangeable products. Okay, and we understood what are challenges behind production of biosimilars, the criteria that are required to be fulfilled for any drug to be biosimilar, and all the basic introduction and importance of clinical trials and all the things that are associated with biosimilars. Okay, I thank you for this enlightening presentation, and we are grateful for your time and effort you took to share your thoughts and experience with our participants. Thank you so much, sir.
Thank you all for patient hearing. Glad to be here and share my thoughts and give you overview. And if there are any other questions which some people want to understand afterward, so you have my email ID. You can collect this question and send it to me. So with that, I thank you. Jai Hind.
Okay. Yes, everybody. Now, after a wonderful session by Dr. Pawar, we have in our midst our second speaker, Dr. Jayachandran Ramalingam. Sir is currently working as an AGM in Serum Institute of India and responsible for the quality control operation for biosimilars. He holds a master in pharmacy with specialization in pharmaceutical biotechnology and is an alumni of JSSCP UT. With around 18 years of industrial experience, he has been an accomplished leader in the area of protein analytical sciences and currently responsible for the complete analytical portfolio for biosimilars. He has started his career with Indus Biopharmaceuticals, Ahmedabad, and later moved to work with USP to lead the biologics lab located in Hyderabad. In his role at USP, he has collaborated with all leading biopharma companies in India and other regulatory bodies across the globe. After a short stint with the Biocon Group, he moved to Serum by 2016. At Serum, he has established a complete analytical portfolio ranging from early stage development, product characterization to demonstrate biosimilarity, stability studies, and eventually to take products to clinical and commercial use. He has received Mrs. Kanda H Gold Medalist for being Outgoing Student of the Year in 2002-2003. He has participated in several national and international conferences as a speaker. With this beautiful introduction, I would like to proceed to hand over the session to Sir. Thank you so much.
Hi, good morning and good afternoon, everyone. Is it audible and clear?
Yes, sir, absolutely.
Let me share the screen. Are you able to see the presentation?
Yes, sir. Yes, sir. It is visible.
Hello.
Hello, sir.
Shridevi? Yeah. Pushkar, are you
Yes, sir.
able
It is
to?
visible. Yes, sir. I'm able to see the screen.
Okay. Yes, I think we are all set to have an interesting discussion for next 40 or 45 odd minutes. Thank you for the introduction. If there is any interruption in seeing the screen or in the audio, please interrupt and indicate if anything is there. So thank you for the opportunity. I am glad that I'm getting an opportunity to interact with the students after a very long time. But at the same time, I would have wished this to be a face-to-face event where I could get an opportunity to come to the college and see those good old days where we have been in college. So Having said that, so let us get into the topic. In this session, I'm going to share my experience as an analytical person, like what are the challenges involved in developing a biosimilar. First thing, to talk about some of the common concepts and some the basic old school definitions about what are biosimilars and what are the critical quality attributes and how in industry one is going over to address all these different aspects, right? In this session today, I'm going to deal with, I'm going to share my experience with how we are intending to develop a biosimilar monoclonal antibody for the treatment of psoriasis. Just to clarify some of the concepts, let us see what are generics and what are biosimilars. I think probably you all must be familiar with the name generics. They are nothing but chemical molecules, which are copycat molecules in a very layman's terminology. They are low molecular weight pharmaceutical compounds which can be completely synthesized in large scale quantities. They have relatively simpler structure, and they can be manufactured in large quantity in a standardized manufacturing process. They are required to demonstrate only bioequivalence. It does not require complicated clinical studies to demonstrate its suitability. Whereas biologics and biosimilars are normally produced in using a recombinant technology, and of course they are high molecular weight compound because they are made up of amino acids. So either it could be a polypeptide chain or having polypeptides assembled into a multimeric molecule. Of course, because of this, they have a complex structure. As I've mentioned, this is recombinantly produced, so this involves utilizing the living cells to make the medicines, and it requires highly sophisticated and specialized manufacturing process skills to make them. And they also involve complex comparability exercise, which we are going to see over in the next upcoming slides over this session. By looking at this, you can understand the sheer complexity that might arise because of the size of the molecule. If you see the aspirin or paracetamol or acetyl salicylic acid molecule, which we all know it has become a household medicine during COVID time. Compared to that, compared to a monoclonal antibody, this is absolutely nothing in terms of its size. If you want to see the impact of its size alone, forget about the complexities added to this. This is several hundred-fold larger in terms of its size of a monoclonal antibody when we compare it with the aspirin. So we are going to talk about challenges or the aspects involved in comparing a biological molecule, which is expressed in a recombinant process, with another biological molecule, which is expressed through a recombinant process. So one molecule is going to be called as biologic medicine because that is going to be the reference compound, and otherwise also it is called as a novel molecule. It is also otherwise called as a registered medicinal product, RMP, normally it is known. And the other molecule is, in layman's term, it's a copycat, but of course that is also a large molecule, conventionally called as a biosimilar medicine. How do we go about it? These are the few of the aspects that we cover in the process of demonstrating the biosimilarity. So it involves a comparability assessment. Reference product, we demonstrate the comparability in terms of its quality, the pharmacokinetic and pharmacodynamic principles, the mechanism of action to be proven, and the efficacy of the molecule, safety. All of them needs to be addressed. We will cover them in the coming slides. Okay, so some old school definitions and some concept clearing points for you to familiarize with biosimilar or biosimilarity. So these are the excerpts taken from, for the ease of convenience, these are the excerpts taken from the public information available from USFDA. So this information are available in public domain. One may want to go through and understand more in detail. There are several such study material available in USFDA domain. So biosimilarity is nothing but the biological product is highly similar to the reference product. And to familiarize with this biosimilar product and reference product, that is where I've shown this slide that, in terms of its molecular nature wise, they are same, they have the same efficacy and safety, but you have to demonstrate that how closely this biosimilar molecule is similar to the reference product. And when demonstrating so, definitely you would have some differences observed. It is not be possible to demonstrate complete similarity. It may have some differences. It may introduce some differences when we are trying to make a biosimilar, but they have to be clinically inactive or no clinically meaningful differences. How do we conclude that? That's a very long topic, and it may not be practically feasible to completely go over all these aspects of what is clinically significant and what is clinically non-significant. But in the upcoming case scenario, we would be going through several such aspects as like live examples. So, in simple words, the biosimilarity needs to be demonstrated for molecules' similarity in terms of its structure and also its safety, purity, and potency, with minimal or no significant clinical differences. And what is a reference product? Which is your benchmark, which is the novel molecule, which is the biological product approved for a specific indication. And normally they are approved through a 351A of the PHS Act. And what is biosimilar? Yes, that is the copycat molecule, which will go through a separate pathway, which is 351K pathway. All these terminologies are relevant to USFDA. I'm just specifically referring to the USFDA mechanism for approving of a novel molecule and a biosimilar molecule here. And when we take the biosimilar molecules assessment and approval, one would take up the 351K pathway, where it is biologically similar, or in terms of the definition, what we have seen in a couple of slides before, biosimilar to a reference product. It should utilize the same mechanism of action. If I am taking a molecule for treatment against psoriasis, it should treat only for psoriasis, only to the extent of mechanism known for the reference product. It should not be used for treatment of psoriasis, even though the disease indication is same, but it should not employ a different mechanism of action. It should target the same antigen, it should target the same receptor, or it should target the same cellular pathway for even for the same indication. The conditions of use proposed in labeling should be previously approved. That means if a molecule is already sought after for a biosimilar pathway for psoriasis, you should have a reference example available approved for psoriasis, and it should have a same route of administration and also the strength. If it is intravenous infusion or if it is a subcutaneous injection, it should be similar in terms of its application. And also we should have the strength of the molecule. Here it is about the concentration and the content given in a vial or an injection. That means if it is an 80 mg or 100 mg per ml, it is supposed to be the same content and quantity in terms of its application also. And definitely all of these products should meet the standards. Here it indicates the global good manufacturing standards, which are conventionally acceptable by various regulatory bodies, and kept on revised as the technology and as the concepts evolve. So they are manufactured and compliant as per the GMP standards and designed to assure the product continues to be safe, pure, and potent. Now we have the process of developing a biosimilar. We also need to understand the critical differences involved in how a standalone, that is a novel molecule, is developed and how the biosimilar molecule is developed. You'll see on the left-hand side of this slide, the goal of the standalone is to demonstrate its efficacy, of course, also its safety. It involves broadly, it involves developing a molecule from scratch, where we are first trying to identify a new disease mechanism, and then trying to figure out what could we do to address this disease mechanism. So we start from a bottom level of the development chain. We identify a molecule which can cater to the therapeutic need, and we first demonstrate its safety in a non-human setup using a relevant animal model, and then study the first-in-human safety using a small number of volunteers, and then study them in actual disease setup in the later stages of the clinical setup. This corona pandemic, I believe, would have given a lot of idea to almost all of the public that what is the clinical development, where a lot of lot of information exchanged and shared over public media, right from TV to internet. What molecule is in what stage of clinical trial, and when the study would be completed, how much data is generated, what number of volunteers were involved. There were a huge amount, enormous amount of information was shared in the public domain. So now, by now, you all might be very familiar with this terminology, such as clinical phase 1 and phase 3. So the late stage trials with a large number of population is called phase 3. In contrast to this, in the biosimilar development, we see that it is the primary goal is to demonstrate biosimilarity, and while demonstrating biosimilarity against a reference product, the other goal is not to independently establish. That means whenever you are doing a trial in an animal setup, or in an animal model, or in a human clinical trial, it is always going to be in comparison to a registered medical product, that is your RMP, or otherwise your innovator product. You are going to compare the PKA, you are going to compare the elimination, you are going to compare the immunological effects. So all of those are going to be in comparison, even in a clinical setup, not just in analytical setup. So in this pyramid would be very critical for any biosimilar development program, where the demonstration of analytical similarity forms the bottom line of this development program, where we are going to discuss in detail, like what are the analytical techniques which are supporting in developing the biosimilarity assessment for a given molecule. And then once the biosimilarity assessment is established, then we go about generating the animal and PK, and then the comparative studies, and also using any other clinical data that may be required to extrapolate the mechanism, extrapolate the approval for other indicated mechanisms which are approved for the RMP. This again has some additional information at the bottom of the pyramid. You see, this is the stage where the design specification is established for both the product and the process. And in this bottom of the chain, the physicochemical characterization and the biological characterization forms a major portion, where the process development and analytics goes hand in hand, which establishes the benchmark for the molecule, and also the benchmark for the product, for the process, because the product process is going to play a very critical role in defining the quality of the product. And once this is established, when we go further for the preclinical and PK studies, then we would validate all the analytical methods, and we would validate the process, establish, streamline all of them, and make them reproducible in delivering the product batch after batch, so that you get a consistent quality of the medicine available once we reach its approval stage. And then the top of the pyramid is, of course, the significant amount of information needed to be generated through clinical trials, of course, in a comparative mode. To summarize all these points, what we have seen so far, the key concepts are: the biosimilarity development is different from developing a novel molecule, and in a novel molecule, it is a standalone product development, where you do not have a necessity to compare it with anything. But in case of a biosimilarity, the name indicates that you have to be similar to something. So it is a comparative evaluation in terms of the product quality and various parameters, there may be acceptable clinical differences which does not significantly impact the effect of the molecule. And the analytical similarity is the foundation of the process. So understanding the relationship between the critical quality attributes and its interaction with the clinical safety and efficacy is very important in determining the residual uncertainty that may lie with the molecule, so that we would be able to confidently justify any minor differences observed in the molecule, does not result into clinical differences. Well, you can practice some tongue twisters given here if you want to practice some of them. So these are not weird things to see. These are the nothing but the names of different molecules which are in development or approved in the market for various disease indication. Why these names are so weird, I have no clue. But what I can explain you is that there is some commonality or some rationale behind naming of these different molecules. Whenever it is, before going to that, you must be familiar with the terminologies such as chimeric, humanized, and human. Chimeric is nothing but it's mixture of two different components where molecule can have a backbone from a mouse, so it can be a mouse component in the constant region, and then the variable component could be a human. And as the technology improved, and chimeric could have its own disadvantages of immunogenicity, things like that. When the technology got improved, we have the provision to eliminate majority of the chimeric portion, and then we made the molecule as much as possible similar to a human monoclonal antibody. But humanized is still having some non-human component in its structure. So human is nothing but a fully human molecule where the antibody developed in a recombinant technology is as comparable as a human monoclonal antibody. Whenever there is a component of XI in the substem of the naming, abscizumab, rituximab, infliximab, these are older generation molecules which are developed using a chimeric technology, and once they are humanized, they are having a substem of ZU in the naming pattern. So they are palivizumab, trastuzumab, and things like that. And then came later generation molecules which are fully human, like adalimumab, panitumab, and golimumab. So practice them when you're alone. We would be seeing an example of how a biosimilar map for a disease indication of psoriasis is getting developed. Psoriasis, we would see the mechanism of action. It is, Stellara is indicated for psoriasis. The Stellara is used for treatment of psoriasis as well as for Crohn's disease. There are two different presentations available. One is 45 mg per ml, 90 mg per ml vial or PFS. This is used for psoriasis, and for 130 mg per 26 ml presentation is used in the Crohn's disease. This is how a very basic mechanism of action and how the disease is developed and how ustekinumab is preventing the disease progression. So the psoriasis involved in various levels of cytokine storming. It's basically an immunological disorder where our own immune mechanism is acting against the dermal cells. Some of the proteins developed by our cells are considered as foreign particles. In a normal setup, it takes 30 or more days to develop the dermal layer. But in this particular disease setup, our whole dermis layer is, coming over within 2 days of time, and the patient develops itches, rashes, and because of all these disorders in the immune mechanism, where our own mechanism is fighting against the dermal cells. So in the normal disease condition, when IL-12 and IL-23 is binding to its respective receptors on the NK and T cells, so the cell signaling pathway is activated. It results in a cascade of cell signaling events, result into cytokine storm, which impacts the dermal layer getting removed and resurfaced in a very short span of time of 2 days. So due to that, the psoriatic indication is formed. And so what to do with this disease? So this mechanism, helps in preventing the disease progression. Ustekinumab binds to P40, which is a common subunit of a dimeric protein, which is IL-12 and IL-23. These are cytokines. So binding of ustekinumab with the P40 prevents binding of the IL-12 with its receptor and IL-23 with its receptor. So there is no cell signaling. As there is no cell signaling, there is no cytokine storm, or there are no immune modulated effects are impacting the cell. So there is a gradual progression of a normal epidermal layer established in over a period of time. Okay, well, this is my favorite part of the presentation. We can talk about it for the entire session, but we will go over about the important components given in this slide. As you all know, this is the structure of a monoclonal antibody. It has 4 chains, two heavy chains, which are the longer ones, and 2 light chains, which are shorter ones, assembled through several disulfide linkages in the orange brackets. They are held together through this disulfide linkages, which gives primarily its structure, 3-dimensional structure. And because of the structure and various components associated with this structure, different aspects of the molecule are becoming critical for the safety, efficacy, and purity of the molecule. Let us go one after one another. So this is called the N-terminal region, and this is called the C-terminal region. Primarily, this is considered as a starting point of the molecule. So N-terminal, whenever there is a glutamine or glutamate involved, it could convert biologically through post-transfer modification into a pyroglutamate, which result into a mass difference. And this different variants can be analyzed through a CAX, CIF, or IEF. So the terminologies, the abbreviations are cationic change chromatography is a chromatographic technique that is used for separating various charge variants. Isoelectric focusing must be familiar to you. And in the structure of the molecule, the primary structure, which is the amino acid backbone of the molecule, it contains several amino acids. Wherever there is asparagine and methionine are involved, asparagine could undergo deamidation in a normal biological setup due to any adverse situation. So this results into loss of addition of 1 Dalton in the structure, thioether formation, and glycation due to excessive amount of carbohydrate source present in the media. When the antibodies are extracellularly segregated by the mammalian cells used in the production setup, this antibody are in contact with the glucose molecules in the media, so it may take up the glucose molecule and get glycated. As we have seen, the methionine could get oxidized, that means addition of oxygen, and the fragmentation could result because of the weaker disulfide bonds. So the molecule can break and form fragments. That means one of the light chain may get loose, or the backbone of the molecule can broken because of the breakage of the hinges. So it may form only a molecule without a light chain, or with 1 light chain, or with only 1 light and heavy chain. Such combinations are possible. So they are also considered as impurities. Other form of modification in the molecule is the asparagine isomerization, that is isoaspartate formation. So these are the different variants that are possible in the molecule because of primarily a complexity involved in its structure. It could be because of the primary, it could be because of secondary. Wherever the asparagine and other things are involved, it is impacting the primary structure. Wherever the disulfide linkages are involved, that impacts the secondary structure. When altogether it is put into a formulated environment, and if that is something significantly changes, it impacts its tertiary structure. And it also can undergo aggregation because normally antibody formulations are in very high concentration, more than 50 to 100 mg per ml concentration, because of closeness, proximity of the molecule. All of them are highly hydrophobic. They could get aggregate and result into loss of its biological activity, or to extend loss of its. It also could initiate or aggravate the immunogenicity when taken in the human body. So different aspects are physicochemical properties and biological properties. Where physicochemical properties are involved, it would impact the primary structure, and this also could attribute to different types of product variants, such as dimer, fragments, deamination, what we have seen in the earlier slides. So the N-terminal pyroglue, C-terminal, and sequence variants impact the primary structure of the molecule. And the biological properties broadly the PAB-mediated functioning and the FC-mediated functioning. So for an antibody like ustekinumab, where we see mechanism involves binding of the antibody to an antigen. So the FAB portion is very critical because the FAB portion is utilized and deployed in binding to a portion of the P40, thereby neutralizing its effect by not binding to its receptor. So this is a target binding or target neutralization, loss of which could result into immunoreactivity or cross-reactivity. And FC-mediated functions, where in the structure of the antibody we have a bracket here. These are glycans. These are nothing but carbohydrate components which are attached to the a part of the heavy chain, which is again required for the stability or the structure of the molecule. They are very critical in mediating the heavy chain mediated function, the FC mediated function. In this case, the glycans present in the molecule can interfere with the FC receptors, which are deployed by the NK cells and T cells. In some molecule where the FC mediated function is also critical, like trastuzumab, where apart from binding and neutralizing the HER2, it also has an ADCC mechanism where the antigen dependent cytotoxicity or CDC, complement dependent cytotoxicity. These are the two different mechanisms that are deployed for cell killing, where there are FC mediated functions are involved. Any deficiency in that would result into loss of its functioning. So in brief, this is the structure of the molecule, and due to various components associated with the structure and its importance, it impacts the several attributes on the functional as well as its safety point of view. And when we talk about, these are some of the techniques we have seen, the STS page, your HPLC, your mass spectrometry, and other techniques, which are some of the tools, but we actually deploy a broad array of tools, a vast array of techniques that are to be deployed for addressing various aspects of the molecule. Some of them are addressing the biological activity. Some of them are addressing the primary structure. These are the technique. This is the domain, and these are the different techniques involved in this. So amino acid sequence, the intact and reduced mass of the molecule, and determining the oxidation, deamidation, and the glycan map. And these are the various points that would attribute to the primary structure of the molecule. And the higher order structure is attributed by the secondary, tertiary, and thermal stability. We also need to, once it is formulated into a vial or an injection, we also need to monitor whether there are any foreign particles or submicron particles which would potentially present a danger to the human body. And also higher order structured aggregates can also result into unwanted immunogenicity displayed by the molecule. And various product related impurities and the process related impurities are listed here. They also need to be controlled, monitored, and also to be compared in the process of biosimilarity assessment. In the process, when we make it in a large scale bioreactors, there are residual protein A, which is coming out of the purification column, residual DNA, which is carried forward by the host cells, which are nothing but the mammalian cells, which are used for recombinantly manufacturing the antibody. So the DNA from the broken down cells could also get into the formulation. And the host cell proteins, which are when the cells break and lyse, the proteins from the mammalian cells also can get contaminated into the media, and that may also get coagulated to the product formulation. So since they are non-human, so they could also get into the product formulation, when injected beyond critical level, they can also pose a threat or risk to the patient safety. In the next, having gone through generic things and the general requirements and brief introduction on structure of the molecule and various aspects related to it, we would see what as a real-time case study or a real-life scenario, what we have done so far for this molecule, what we are developing in-house, which is intended to be treated in a psoriatic indication. So when we make this product, first of all, we need to first determine and qualify whether this molecule what we are making is first of all of adequate quality before in fact we actually take it up for broad scale comparison with the biosimilars. We have a benchmarking called a release specification, which is used for first to qualify the molecule whether it is good enough to be considered as a quality. So these are the various parameters which we employ: the appearance identification and the determination of the impurities by size exclusion and the purity by CSDS, and also the binding of the potency of the molecule in terms of its binding affinity to its receptor using ELISA based and intracellular cell based assay in vitro setup, and some of the process components are quantified. So these are the basic set of activities we do to say that yes, this molecule meets a skeletal requirement and it is considered to be of adequate quality. Once we see that, then we generate multiple batches of this molecule, and using this benchmark as the lot release criteria, we call it as a lot release criteria. That means a batch of a product is passed or failed using this as a benchmark. Once it is established, then we make multiple batches in a large scale, and then we test them, and then we say whether it is complying to the requirement or not. And we monitor, for example, the identification in terms of PI, that is isoelectric focusing, is kind of an identity for the molecule. Is all my molecule is same, so I quantify the PI and it is giving me the same PI, and it is consistently same, so it complies, and the pH in the formulation is same. The osmolality, which could significantly impact the patient safety, is also same. And critical quality attributes, whatever we have defined, are monitored, quantified, and certified that yes, they are of course same and within safe limits. If you all notice that there are quantitative results, and you should not be surprised that the numbers are varying from batch to batch. So identical or similar does not mean that you would be getting 51.83 for every batch is next to impossible, where we have a defined criteria of upper limit and lower limit. And within the limit, the product is supposed to behave in an expected pattern. So that is where we call it as acceptable quality. So similar or identical does not mean that every time you get a protein concentration, you would be getting for all the batches 95. No, that's not the meaning of similarity. So I hope this slide could help in clearing some of your concepts. That similarity doesn't mean that having the same thing for everything is actually next to impossible. And these are the different aspects: the bacterial endotoxin and sterility. Of course, when you are taking some injection, it should be sterile. That means it is free of foreign material. Foreign microbial organism. Otherwise, instead of getting the treatment provided, you would have to treat it for some other aspects. And once this drug I will also like to clarify a concept here. This is the benchmark for the drug substance, and drug substance nothing but it is manufactured in large quantity, available as a bulk material, and when they are filled into a vial or an injection, they are called a drug product. So when we fill into a drug product that is ready to administer into a human, whether it is for a clinical study or for a therapeutic when it is approved. So this particular presentation is called a drug product. In drug product, we have a different criteria to assess. That is, presence of foreign particle. You should not see any particulates. You should get an expected delivery volume when you're injecting 1 ml. You should get 1 ml, and it is not colored or it is slightly colored within a controlled level. It should not have any foreign particle in terms of your clarity. And of course, the identity, purity, potency, everything is established. Again, you have to demonstrate the absence of endotoxins, which would induce unnecessary immune reaction, and it is sterile, does not have any microbial contamination in the product. Once it is done, we would take up the stability study, because before taking it to the market, we should assess and establish, yes, the molecule what we are making is stable and it is going to lie in the shelf for 12 months to 24 months. And after that, at least until the end of its shelf life, it is suitable for its intended use. So we apply various levels of stability studies in a normal storage condition, in an accelerated storage condition, to study the effect of its transportation on the product. So these are the various stability studies we cover. The long-term studies for storing in the actual storage condition, the accelerated condition and stress condition, are extended temperature ranges to see the impact of the temperature on its storage and its efficacy. For example, here when we study the DS, that is a drug substance, we have studied this molecule for 12 months. This is only a very preliminary data. We normally study up to 12 to 24 to 36 months time. And we have to demonstrate that all the levels, all the parameters what we are observing are within the set limits or acceptable levels of upper and lower level criteria defined for each of this parameter. So we study the DS for its suitability, and we also study the DP for its suitability. DP is studied for its stability in the given packaged condition. That means it is in a vial for infusion or in a vial for injection, or it's in a pre-formulated syringe, things like that. And then we go about with the characterization. Once we have this kind of a benchmarks established for the molecule, we have to then venture out to characterize the molecule. I have taken a gradual steps how this enters into a characterization. Sometimes the characterization, I do not want to go into the the elementary levels of characterization. Sometimes the characterization happens at a very small scale level to say that the molecule has a defined criteria. And also when it is going towards the late stage clinical trials, we deploy large number of batches for comparison. We also deploy broad range of techniques before we actually take it to the regulator. So it is also having various stages. But here in this case study, I just want to cover what are the techniques we have deployed for characterization, and the requirement will definitely change over a period of time. When we take this study to a regulator at a very, very early stage preclinical trial, the requirements are different. When we again go to the regulator for getting an approval for a clinical trial application in a phase 1 study, so the requirements are different and the extent of characterization is different. And at the same time, once we have done all the characterization, now we want to take it to the market. We want to apply for a commercial approval to be marketed as a approved formulation. There, the number of batches to be compared and the extent of parameters to be compared is going to be different. But what is to be understood from this few slides is that this is a basic mechanism of how what are the critical parameters involved in the characterization and how the characterization data is generated. So if you see here, the amino acid sequence or its primary structure is evaluated by peptide mapping, intact and subunit mass analysis, and the secondary and higher order structure through a near and far UVCD fluorescence spectrum, the disulfide mapping by non-reduced peptide mapping, and the differential scanning colorimetry. The aggregates are evaluated by SEC and multi-angle laser light scattering, SECMALS. The glycoform variants are analyzed for various aspects, which are the non-glycosylated species. That means the carbohydrate backbone is absent in the molecule, and these are the different category of the glycan components in the molecule. The oxidation and deamidation are impurities. The charge variants again for part of the product-related impurities, and the functional aspects, the Fc receptor binding and the target binding activity using various assay setups. Now, this is the profile of peptide mapping, where we take the molecule, we reduce them with the DTT, we break them apart, and then apply an enzyme, normally typically trypsin, to break them. You know what trypsin does to an antibody. It is a proteolytic enzyme. It breaks down the peptide at a specific region. So the molecule is broken down into very, very small pieces, small amino acid fragments, and then this is studied in HPLC and visualized to see that the pattern what you get, the red color indicates the pattern of a standard. Here you have to recall whatever we have discussed in the initial slides, which is the reference product. Right? So it is normally a biological medicine which is approved for its intended use, and it is already available in the market. We use them as a reference product, and we treat them in the same way how we want to analyze our product. So it has to give a profile, and not necessary to say that they both have to give you the expected profile. So then you see that they are comparable and similar. So this is an example of a peptide mapping profile where you evaluate the biosimilar molecule for its similarity with the reference product. And then we use mass spectrometry to evaluate the composition of the molecule, the different peptide peaks which are eluted by chromatography are subjected to mass spectrometry, where each of these components are identified, each of the amino acid sequence of this peak is identified, and then we collectively collate everything together and see that whether the sequence theoretically published for the reference product is obtained, and then we also match for our molecule the same sequence what we are getting. So if we have the 100% sequence coverage for both the standard and sample, we can confidently say that yes, the product is same as what has been licensed by the regulator. So this also determines its identity of the molecule. And then for the intact mass, we also study the intact mass analysis, and this is the mass of the intact molecule. That means the molecule as a whole. We using LC and MS, we study the mass of the molecule, and this is the theoretical mass, and we get absolutely similar. I must emphasize the word similar. It is not identical. It is similar, and it is within 50 ppm levels of variability, which is normally considered acceptable for the molecule. And these are the different backbones identified in the molecule. They are similar within reasonably acceptable variance for the molecule. And this is for the again for the deglycosylated. That means to avoid the variability, these are multiple peaks obtained. Now if you want to eliminate the variability, we deglycosylate. That means we take out the glycan from the molecule, and then we study them again. So it gives you a single component, and the only variability observed is because of the N-terminal and C-terminal region, where the N-terminal could present in a pyroglutamate formation, and C-terminal could present in a clipping of lysine variants at the C-terminal. So it gives us, almost same levels of mass value for all the different species. And then we break down the molecule. We study the subunit, light chain and heavy chain. And here is the detailed list of different species identified. Light chain will not have any variant, but the heavy chain will have a glycan and backbone variants. They are also identified and compared. And we again study them in a deglycosylated condition. That means the glycan is removed. So we see the light chain, heavy chain, and the protein backbone identified and compared within reasonably meaningful levels of variability. And we see the structural study comparing the secondary and tertiary structure. Near UV CD is nothing but a circular dichroism, another tool used for evaluating the secondary and tertiary of the molecule. So in the near UV CD, we see the tertiary structure of the molecule. You can get back to your biochemistry books to see what is the secondary structure and the tertiary structure. Secondary structure basically constitute the alpha, beta, and random coils formed due to the peptide backbone association. And the tertiary structure is the three-dimensional structure associated with the molecule, particularly when the molecule is very large, and also having multiple peptide chains in its structure. So overlapping of all these different molecules into a same line indicates that they are considered to be similar in terms of its secondary and tertiary structure. And again, another tool for fluorescence spectroscopy determines the intrinsic fluorescent property of the molecule. If it exhibits the same excitation spectra, emission spectra, it indicates that the molecule is having a similar three-dimensional environment, which is confirmed of its similar tertiary structure for the molecule. And this is in a non-reducing condition where we do not use the DTT, but we still break the peptide. So because of we are breaking the peptide with the trypsin, the disulfide linkages are still open. So the light chains and heavy chains are still held together. And now in this study, we can see the wherever the light chain and heavy chains are linked together. So this is an indirect indication of your the molecular integrity, that all the disulfide linkages associated with the molecule are intact, in fact the same, and the profile match indicates that the innovator's product, that is the reference product, and the biosimilar product have a similar disulfide linkage pattern. And this is a table summarizing like what are the different linkages associated with the various batches, and all the linkages are identified and confirmed positively. Aggregates, the name indicates association of two or more molecules to form a non-covalent association. This is posing a threat to the patient because it may introduce immunogenicity or loss of potency. The pre-peak, that's a small shoulder before the main peak, is the peak for aggregate, where they are quantified and assessed. They are supposed to be similar, expected to be within the acceptable levels of variation, and using the multi-angle laser light scattering technique, we have assessed the average molecular weight, and also we have assessed the polydispersity of the molecule and considered to be acceptable. And using capillary electrophoresis in a non-reducing condition, we have assessed the different fragments that are possible in the molecule. This is the position for the light chain variants. This is for the heavy chain variants, and this is a different impurity. And we have two heavy chains only assembled, and this is a molecule with only a two heavy chain and one light chain. One of the light chain has been lost. So these are the different combination for the fragmented structure formation for from the ideal setup of four chains, that is two heavy chain, two light chain. If that is disassembled or misassembled, these are the different combinations possible, and they are separated by capillary electrophoresis and then quantified. So these are the different impurities identified for the molecule. And glycoforms are nothing but the carbohydrates present in the molecule, which are essential for its various aspects, the stability of the molecule, and also for its FC-mediated mechanism. We quantify the antibodies which are not having glycosylation. They are quantified as non-glycosylated impurities. And we also study the different carbohydrate moieties attached to the heavy chain. Using the LC and MS, we identify various fractions, and these are the different species identified in a given molecule, and they are matched for its presence in the innovator in the biosimilar molecule. This table is for the list of peaks identified for an innovator, and this is the list of different peaks identified in a biosimilar compound. And then we apply some calculation to see that how much is galactosylated, how much is not having a fucose, and how much of a manipulation. I will not go into the significance of what is the importance of galactosylation, what is the impact of manipulation here, but these are the different category of the glycan variants present in the molecule, and they are significant for one or the other reason, and they are compared with the innovator. So wherever you see in the table, I'm not emphasized on this. The RMP is nothing but the innovator's molecule. We have compared multiple batches of innovator's molecule and some batches of Innova's product. And this is the quantitation of a sialic acid present in the different molecules. And the charge variant, the name indicates the overall charge of a molecule could differ, and because of that, it could result into loss of potency or aggregation. And there are different category that leads to. There are acidic variants, that means they are acidically charged, that means having lesser than neutral charge, they are more than neutral charge. So the main peak is the peak of interest. If you see the ones which are coming after the main peak. The basic impurity in this case, the one which is coming before the main peak is the acidic impurity. I'm sorry, I'm telling it wrong. So the ones which are before the main peak are the acidic variants, and the ones which are coming after are the basic variants. And we characterize that the basic variants are indeed because of lysine variants, which are not post-translationally not clipped, and that is because this results into a variant. And the functional studies are addressed through various binding kinetics and ELISA and other in vitro mechanism. We apply Biacor. It is a surface plasmon resonance is a mechanism which study not just binding of the molecule between the antigen and the antibody, but the study reveals the affinity of the molecule. That means how tight or how strong is the affinity of the molecule towards this antigen. The stronger the binding affinity, the better the efficacy of the molecule, or the better the impact of its molecule in neutralizing effects. So this is using IL-12. As we have seen, IL-12 and IL-23 are the two target mechanisms for neutralizing for psoriasis for this molecule. So we study the affinity of the antibody to bind with IL-12 is a KD value demonstrated through a KD value, and the affinity to neutralize the IL-23 is demonstrated through the KD value here. So the lower the levels indicate that they are very, very strong binding to the intended receptor. Since we have this P40 is the common subunit between IL-12 and 23, we have also separated the P40 and studied the binding mechanism, and then we have proven it again. So these are all orthogonal tools to prove a point that yes, they have indeed a highly strong binding capacity to the target antigen. And we have also applied a cell-based assay to say that it is not just having the affinity at the in silico or in vitro setup. We have also studied it in a cell-based, relevant cell-based model. We have demonstrated the cell-based assays to compare it with the innovator's product and demonstrated the potency of the molecule. And we have also employed the ELISA-based assays for determining its potency. And we have also some aspects of FC receptor binding. As we have seen, FC receptors are critical for demonstrating the FC-mediated functionality such as ADCC and CDC. So this demonstrates the affinity of the antibody towards the FC receptors. So these are the FC receptors used, FC gamma 3A and the FcRn are available as a reagent. We bind the antibody with this receptor and study the binding affinity of the molecule. Overall, this is the summary out of all the list of tests we have seen earlier. What are the parameters I am testing and by what method I am testing. Now I have added a component that how significant are the results. In some cases, I would say they are identical. In some cases, I am saying comparable. And for example, here in case of acidic variants, particularly, charge variant. So I say that there are some differences, but that is because of inherent variability in the molecule. The basic variants are less in our product, but it is most in the innovator product. But because of some principal mechanism involved in the way how the cell lines are, the antibodies are manufactured from different cell line. So we would substantiate with significant justification, with relevant justification to substantiate how this difference does not constitute clinical differences. If you see in our, if you remember in our earlier slides, we mentioned that it should be demonstrated similarity, but of course they should, it is expected to have some difference, but that should not be demonstrated clinically non-relevant. So in a nutshell, yes, we would be making a molecule, we would be comparing it with a wide array of tools, and they are really very broad set of analytical technologies, highly complicated. We may get exactly the same result, or we may not get exactly the same result. Then if it is something different, then we will have to investigate whether it is clinically significant or not. And if it is not significantly different, then we would substantiate it with other orthogonal techniques and or other studies that we may want to provide and justify to the regulators to accept our molecule as biosimilar to the licensed product and would get get it approved. So, so this is what I have tried to put it in a nutshell. The very brief glimpse of what is happening in the analytical setup, because everybody is talking about analytical setup is the bottom of the pyramid. You have seen that already. So this is in a very nutshell. Again, it is very complicated. It is not as simple as it looks like. But in a nutshell, this is a glimpse that you want to know, or you want to see that what all happening at the analytical setup. At the end, I want to appreciate the college initiative that uh, the college is taking initiative in introducing the topic of biosimilarity or the biosimilar development at a UG and PG level, which is I'm really very happy to see, which I have started witnessing, or at least I have started learning only after getting into industry. But I'm happy to see that the graduate and postgraduate students are getting an opportunity to at least get familiarized with the concepts associated with the development and its characterization and the overall biosimilar pathway. I'm glad about that. So I conclude my presentation, and it is open for discussion now. Thank you.
Okay, sir. We have questions. Okay, there's one question which says, is there any library for biosimilars?
I'm sorry. Can you?
Is there any library for biosimilars?
You mean a list of molecule which are approved already? Is that what is the meaning of the question? If that is the meaning of the question, if it is a library, means the regulators, whether it is Indian or US FDA, they publish periodically the list of biosimilars approved by them for use as biosimilars. But if that is not the meaning of the question, if you ask me, what are the biosimilars that can be developed? Again, it is purely depend upon the company's interest. The companies normally identify the potential high business value molecules, which are biological medicines, which are approved already by regulators, either by USFDA or by Indian regulator. And if it is having a high business impact and also significant disease burden, if a company wants to address that as a disease burden and also take it as a business case, then the company would identify that as a target for developing a biosimilar, and then would go over about developing biosimilar.
Okay, sir. There's another question which says, how much difference in the glycosylated intact mass of the reference and similar makes it validated?
How much difference? There is no hard and fast rule. We want to have the difference as minimal as possible in terms of glycan variants or non-glycan variants. At intact level, we want to have it less than 100 ppm at 145 kilodalton. Yeah, 145,000 dalton. So we wanted to have it is as a thumb rule. Want to have it as less than 100 ppm. At a subunit level, we want to have it less than 50 ppm. These are the practical levels we are trying to maintain.
Okay. Which is the most prominent characteristic or a criteria which will help reject biosimilarity?
No, there is no single criteria. There is no single criteria that you can pinpoint and say. If you have to, blindly accept or reject the biological activity is the most prominent one. But I would say it is not the only criteria. If you see all those parameters which are listed in the table have significance, and that is the reason all of them have to be complied, either very closely or absolutely safe.
If you would compare the time period required for characterization of the reference and then your product, is there a time variation between it for complete characterization or a comparative analysis between both of the products?
See, normally comparative analysis means it is taken up during, for example, if I'm studying size exclusion chromatography, I am going to analyze both the molecules together. in the same analysis. So these multiple analytical techniques that are listed there, they are in standalone. They are tested for its effect in comparison to an innovator. So it is always studied in comparison to an innovator in the given analytical setup.
Is the biosimilar for cirrhosis cost effective?
I'm sorry.
Is the
Can you
drug that is created is cost effective?
Cost effectiveness is a different angle. Definitely, biosimilars will be cost effective because the amount of IPs they are creating, the innovators are creating, the IP means the intellectual property ownerships they are creating, and the amount of time and the cost they are investing in the large scale clinical setup and demonstrating the proof of concept, and the time they are taking to develop this molecule is significant. It runs into billions of dollars. And when we have different set of challenges. We are not working in standalone. We're always working in comparison. So, but the cost of production does not change. We have the same GMP setup, and we have to make it in large quantity. We need to have large scale bioreactors. So the challenges associated to its manufacturing is not significantly different. But since we are avoiding some of the clinical studies that the innovator is doing, so because of that, we are biosimilar development is definitely less costlier than developing a novel biologic, and that
What
cost difference can go to the patient, where it is expected to have biosimilars are available to patients with a lesser cost than the novel biologics.
what according to you is the main risk associated with using of biosimilars?
Once it is approved, there should be no risk. Actually, there is a big debate on interchangeability. That means if a doctor is writing, it is there in small molecules also, but that is well demonstrated, and you have well established practices available for interchangeables. In Lerman stamp, if you are being written with Palpal, you can buy something else which is containing paracetamol. Do you see there is any risk? I don't see there is any risk. So applying the same logic, once the biosimilar is evaluated by the regulator and approved for its commercial use, there should be no risk posed to the patient on its safety point of view.
How are the biosimilars for cirrhosis tested?
You mean the analytical testing?
Ms.
Okay,
Devyanshi,
if you
can
mean
you elaborate on your
yeah,
question?
yeah, if you mean the analytical testing, this is where we have actually gone through this presentation. I have listed down the various tools. Some of them are platform techniques, some of them are novel analytical techniques. I won't be able to go through the clinical requirements because it should be part of a separate discussion. I won't go about any clinical development plan for this. But if you're asking me the analytical development and the analytical character, this is what we have seen the last 1 year. So all these things were applied for this.
Do we have any more questions? If there are, please post the questions.
Any further questions?
No, sir. I guess there are no more further questions. I will just go ahead and thank you. Okay, I wish to express my gratitude to Dr. Jayachandran for providing an insight on the topics of chemical and analytical strategies in the process of putting light on the process of biosimilarity or biosimilar development. Okay, talking about antibody structures and their assessments and characterization and comparative analysis. Thank you, sir, for your insightful presentation. We are grateful for your time and effort. Okay, you took to share your thoughts and experience with our participants. Thank you
Yeah.
so much.
Thank you. Thank you very much.
Okay, we do have a question. I guess Saras has left. Okay, Saras left. Devyanshi was.
Good afternoon to one and all present here. I, Shweta Patel, welcomes you all to the second half of RUSA sponsored national webinar Biofacet Biosimilars. Let's begin this session with an introduction of our next speaker, Dr. Ajmera Ramakishan. Sir has done PhD in pharmacology from Gujarat University and is currently working as Deputy Drugs Controller, CDSCO, DGHS, under Union Ministry of Health and Family Welfare East Zone, Kolkata. He is instrumental in enforcement of Drugs and Cosmetics Act 1940 and Rules 1945, thereunder to ensure quality, safety, and efficacy of drugs, cosmetics, and medical devices, and diagnostics, laying down standards, making amendments, approval of new drugs, clinical trials, and monitoring of import and export of drugs, inspection of pharmaceutical manufacturing units for grant of GMP certificate as per WHO guidelines, and also for the inspection of pharmaceutical units covered under CLAA scheme. He also served as an expert committee member of Indian Pharmacopoeia Commission, National AEFI Committee on Vaccines, and member of National Pharmacovigilance Program of India. He has received the Best Appreciation Letters from Drugs Controller General, CDSCO, Ministry of Health, Government of India, for outstanding contribution in the field of biological products and NRA Assessment 2017, and also for favorable judgment to Union of India with respect to simplification of regulatory pathway for approval of biosimilar in India. He served as conveyor in preparation of Biosimilar Guidelines 2016, published by CIDESCO with the support of Department of Biotechnology. He has 23 years of regulatory experience in National Drugs Regulatory Authority, that is CIDESCO, and involved in various policy-related meetings for development of guidelines related to GMP, GCP, GLP, GDP, clinical trials, BA, BE studies, post-approval changes of biological products, pharmacovigilance of immunoglobulins and biosimilars, etc. Serving as member of WHO DCVRN, and also serving as conveyor in subcommittee of Drugs Consultative Committee for preparation of formulation development guidelines and good testing procedure guidelines of various government drug testing laboratories in respect of sampling, testing, and drug test reports. He has earned total 32 awards during his career. Listing few of those: recipient of Guinness Record of the World Certificate 2019 for the largest oral hygiene lesson, and mega medical cancer screening camp conducted for oral cancer in a single day of 5,478 persons held at Telangana state. Then recipient of Dr. B.R. Ambedkar Ratna National Award 2019 for excellent services in regulatory affairs by Karnataka Bahujan Academy, Bangalore. He has been awarded the prestigious fellowship of Andhra Pradesh Academy of Sciences 2019, Fellowship of Indian Pharmaceutical Association 2018, Fellowship of Indian Pharmacological Society 2015, and Fellow of Telangana Academy of Sciences. He has received Best Drug Inspector Award 2005 by All India Drugs Control Officers Confederation. Recipient of appreciation letters by CIDESCO for outstanding performance in NRA WHO re-benchmarking assessment 2017 carried out by WHO Geneva along with 15 countries' regulatory experts. He has obtained 2 patents, 28 research publications, 8 international and 18 in Indian publications. He has also published 2 books in pharmacy. He is also serving as a supervisor for guiding MPharm PhD students, and he has delivered around 107 lectures on drug regulatory affairs in various international and national seminars. He is also a member in various ministries of expert committees under drug policies under central government. His main motto is service to humanity. It is a great honor to have such an eminent personality with us here today. I would now request Sir to please take over the charge. Thank you.
Thank the organizing committee for inviting me here today to give the overview of biosimilar regulations in India. So, as a regulator, how we have to review and what sort of important parameters should be taken into the consideration with respect to approval of biosimilar products in India. So I would like to give the important outline of my today's presentation. What is mean by biosimilar? How the biologics are playing significant role in healthcare sector, and what sort of challenges we are having. So, as a manufacturer, how we can focus with respect to generic version product when the reference products are going to be patent off. So, and what are the difference between the reference product and biosimilars? How we can go for the important physicochemical characteristics and analytics with respect to functional analysis, and important characteristics with respect to pharmacokinetics and pharmacodynamics, and we can have one by one. So this is very important, and yes, next feature is the biological market and how we can go further. So can I have next one, please? So these are all important things, and how many authorities are involved with respect to the licensing of biosimilars in India, and how the regulators are taking play key role with respect to the scrutiny of common technical documents, how the DBT RCGM department with CDSCO, the licensing part, how they are scrutinizing with respect to the preclinical approval and clinical trial approval by the competent authorities. Then what is the data required for the preclinical studies, data requirement for the clinical trial application, and conditions for waiver of safety and efficacy, and post-marketing data for similar biologics, and key change in the DBT and CDSO guidelines 2016. So, in nutshell, you can understand what are the basic differences between the important requirement for approval of market authorization of biosimilars in India. Next one, please. If you see the background, the Department of Biotechnology, through a review committee on genetic manipulation, is overseeing and approving the development and preclinical evaluation of recombinant DNA-based products in India. So, what is CDSO? CDSO is Central Drug Standard Control Organization, is the national regulatory authority, and takes care of the evaluation of safety, efficacy, and quality of drugs, including similar biological products. Then the state licensing authorities and central licensing authority both involved as per the central licensing scheme with respect to approval of drugs under different form. That is, Form 28D is the main license for issue of such category of biosimilar products in India. Next one, please. So, if you see the international design, what is mean by the biosimilar and how we have to take into the consideration? Simply, if you can see, biologics are, everyone knows, novel therapeutic products, and simple. It is not that simple. There are all complex proteins involved and all important technologies involved with respect to development of product. So if you can see, biologics minimum 15 years it will take to develop, and cost is huge, 1200 million USD dollar, and the reference biologics are going to be patentable, and they can have price whatever they want. So that is the important thing. And when the biologics are going to be patent off, when it is going to be expired their patent exclusivity, then the generic version products are going to be developed by the manufacturers, which takes 5 to 7 years time, and whether the safety and efficacy, pharmacokinetics and dynamic parameters are comparable with respect to the reference biological product is nothing but a biosimilar, and compared to that, the cost is very less, 100 to 200 million USD dollars cost and non-patentable, and you can reduce the price. So, being a huge populated country and lot of biosimilar companies, almost more than 36 molecules approved in India since last 6-7 years. If you can see, our manufacturers also extremely competing with global market, and we are having good players now in the market for the development of generic version products, so-called biosimilars. The originator and biosimilars, we can see only two steps involved with respect to the development of biosimilars. Step one is preclinical, so that takes care of the complete characterization with respect to the analytical part is concerned, that is structural and functional analysis, process development, in vivo studies, including toxicity, all important toxicity studies as per the Schedule Y requirement of our Drug and Cosmetic Act, and pharmacokinetics, pharmacodynamics, including immunogenicity studies are very, very important. So all these preclinical studies data is going to be reviewed by the RCGM, important committee, review committee on genetic manipulation, comes under the Department of Biotechnology, that is Ministry of Science and Technology, Government of India. So once they review the data submitted by the manufacturer, if data finds satisfactory, then meeting the requirement of safety and efficacy parameters in animal models, then DCGM, that DBT department, RCGM, apex body will give the NOC to approach the Office of Drugs Control General India Office, that is CDSCO. Thereby, they can make an application to carry out the permission from phase 1 to phase 4 clinical trials in human beings. Then CDSCO is going to take lead with respect to the submission of manufacturers of the respect to preclinical data of respect to biosimilar product, and whether it is comparable and complete characteristics analytical part is going to be reviewed in common technical document. Then whatever data is submitted by the manufacturer, we will review. Then we will refer it to subject expert committee. Subject expert committee consisting of all biologists, pharmacologists, microbiologists, biotechnologists, and physician, and specialized subject, specialized product expert in that important molecule, we also sometimes involve. So then based on the recommendation, if data found to be satisfactory, then they will issue the necessary NOC to carry out human clinical pharmacology. And once they give the approval, then manufacturer has to carry out human trials from phase 1 to phase 4. If it is applicable for phase 4, then otherwise phase 3 data is adequate enough to get the market authorization. So this is a normal routine path involved for the approval of biosimilars in India. So this is the only slide just I want to share. Next one, please. Next one, please. So you see the current statistics, the important inputs I got from DBT, and if you can see, and BIRAC people also taking lead with respect to the promotion of all important biotechnology manufacturers in India. They are promoting all young entrepreneurs, young scientists to focus on novel molecules, because of lot of patents are going to be off in near future. So at affordable cost, if biosimilars are in clinical practice, then at a reduced price without compromising safety and efficacy parameter, we can treat all important diseases, particularly cancer and wide variety of important diseases and important other CNS disorders we can take care. So these are very, very important. And this, if you can see, India has got more potential. If you see in vaccine sector, we are number one with respect to diphtheria, pertussis, and tetanus, measles, and BCG vaccine. Second highest number of USFDA units in India. Third largest biotech destination in Southeast Asia Pacific region, and we are having 3% share in global biotech industry, and 39 biosimilar products approved in India, 3,500 plus startups, and 100 biotech incubators, and 250 billion USD biologicals market by 2030. So these are all important things. And biologics are nothing but what are biologics? If you can see, an approved product composed of proteins, nucleic acids, or combination of these, are living entities such as cells and tissues, which is isolated from natural sources including humans, animals, and microorganisms, and produced by biotechnology methods and other cutting edge technologies, are nothing but biologics. So next one, please. So definition, if you can see the definition, because earlier we were not having any biosimilar definition in our document, CDSO document. First we have prepared in the year 2008. After that, revision took place in 2012, and in the year 2016, a new version of CDSO guidance document with the support of Department of Biotechnology, DBT, New Delhi. We had very good guidance document for approval of market authorization of biosimilar products in India. So what exactly biosimilar? First time we have incorporated the definition of the biosimilar in line of USFDA, in line of WHO Geneva, in line of EMEA guideline. So if you can see the definition part, the biosimilar is nothing but a similar biosimilar is a similar biological product is that which is similar in terms of quality, safety, efficacy to an approved reference biological product based on comparability is called as a biosimilar. And what are the important principles incorporated in that biosimilar products? So if you can see, friends, main principle, approved innovator biological product is very, very important. We have to see whether it is approved in ICS countries, approved in India, or approved outside of India. How many countries really taking care with respect to their distribution and marketing part. Then reference biological product should be licensed in India or ICS countries and shall be an innovator product. Then reference biological product license based on full safety, efficacy, and quality data. Dosage and dosage form, route of administration of biological substance should be the same. Active ingredient of biological substance product must be similar to the reference biologicals. Similarity by extensive characterization, so it should meet head-to-head comparison is must. So it should not be any difference, and it should be comparable with reference product. So they are all important principles incorporated in our existing guidelines, friends. Next one. So applicable regulations and guidelines. If you can see the important guidelines, Drugs and Cosmetic Act 1940 and the rules came in the year 1945 as amended from time to time. Rules for the manufacture, use, import, export, and storage of hazardous microorganisms, genetically engineered organisms or cells, published in the year 1989 under the Environment Protection Act 1986. So recombinant DNA safety guidelines we got in the year 1990. Guidelines for generating preclinical and clinical data for recombinant vaccines, diagnostics, and other biologicals established in the year 1999. CDSO guidance document came in the year 2008. After that, reviewed in 2012, and again important guideline came in 2016. So they are all important statistics with respect to the regulations and guidelines for approval of biosimilars. Next one, please. So, what are the important biotech products approved globally and approved in India? If you see the top 10 biotech products, adalimumab, trastuzumab, all the 1 to 10, if you see, they're all important novel molecules and how they are playing a significant role in healthcare sector. So, adalimumab for the treatment of rheumatoid arthritis and several other indications. Trastuzumab, breast cancer, and gastric ulcer cancer, and other early metastatic breast cancer. So this is one of the top product. Trastuzumab is just taking care only $4 billion annually. So very life-saving and highly potential product, and there are all things so you can understand in nutshell. So for the easy remembrance, and students and young entrepreneurs can understand the importance of the biotech products evolution. Next one, friend. So, growing biological business, oncology, it is covering almost 40%, CNS 16%, metabolic disorders 12%, anti-infectives 8%, and others we are taking care around approx 25 to 30%. So, this is the statistical information with respect to the growing biological business and patent expiration to offer biosimilar business 2022 and 24, 26, 28. If you can see, these are all important molecules are going to be expired in near future. Then our entrepreneurs can be ready for their development of the generic version product, that is biosimilar, and which takes 5 to 6 years time, 5 to 7 years, whereas in case of reference biological it takes minimum 15 years. So you understand you can reduce this cost, and you can have affordable product for the benefit of poor needy people in international commerce. Next one, friend. So these are all competent authorities that I already covered: IFBC and RCGM and GAC and CDSU. So CDSU is the apex regulatory body, comes under the Ministry of Health and Family Welfare, Government of India. And the review committee RCGM, with the close coordination with CDSU, we are taking care with respect to the preclinical data review. Once the data is reviewed, then they will refer it to our office for approval of important clinical trial permission. Next one, friend. So this is the flow, the drug approval process in India. So whenever you go for import of strain, or whenever you are ready for the development of your product process, then strain is very, very important. Then the RCGM, DCG and FDC approvals are required with respect to your development of the product. So once you develop the product, then you have done your animal model studies, preclinical studies as per the Schedule Y requirement of Drug and Cosmetic Act. Then the data will be reviewed by Review Committee on Genetic Manipulation, that is RCGM. Then once the data is satisfactory, then preclinical approvals are going to be issued to the respective manufacturer to carry out further in human pharmacology is nothing but clinical trials. Then they have to approve the DCGI. Then when they go for the grant of drug license in Form 28D, then state licensing authorities, with the support of central licensing authority, they will carry out the thorough audit. Then they will give the approval for facility, what we call manufacturing unit of the respective product, propose the biosimilars, then approval takes place, then approval of drugs in the market, then it will go. So these are all things. So this is the process flow with respect to drug approval in India. Next one, friend. So these are all the basic differences between the reference biological products and biosimilar similar biologics. So reference biological, so you can understand the RCGM requirement, IBAC approval, IAC, toxicological studies after RCGM approval. So you can take care how we can have the basic differences with respect to data for preclinical submission. Next one, friend. So preclinical studies, so pharmacodynamic and toxicology study. If you can see the pharmacodynamic studies, in vitro study, that is cell proliferation, assays, cytotoxicity, neutralizing receptor binding assays, And in vivo studies, if in vitro assays are not available, then they have to go for in vivo studies. Toxicology studies in pharmacologically relevant species as per the Schedule Y requirement of Drug and Cosmetic Act, they have to take care. Immune responses in animal model, antibody response as compared to reference bar. So they are all things, but when they when the manufacturer is ready, when the preclinical data is submitted, then one application can also parallelly they can submit to the DCG office for preclinical to clinical trial permission. When they want to go, then they can take care. So parallel submission and parallel submission acceptance we are having. So one copy they can submit to DCG office and one copy they can submit to RCGM. Parallelly they can review in order to save time, and we can reduce the time then in order to promote the entrepreneurs for fast track approvals with respect to their clinical trial permissions in India. Next one, friend. So there are all things. So as per the CDSO guidance document developed in the year 2008, the quality comparability data, and there was no difference in critical quality attributes, clinical trial permission CT06. So that is the application number. And phase 1, phase 2, phase 3, phase 4. So there are all the phases everyone knows. Phase 1 takes care of the safety and tolerability. Phase 2 takes care of the effectiveness, side effects, risk and dose range studies. Then dose used in phase 2 clinical trials is less than the phase 1. Then demonstration of the therapeutic benefits when they go for phase 3. Then you have to show the evidences with respect to efficacy parameter when they go for approval. And post-marketing surveillance is nothing but phase 4. So these are all different stages of clinical trial permissions undertaken in India with respect to the biosimilar products. Next one, friend. Next one, please. So condition for waiver of safety and efficacy study. So structural and functional comparability of similar biological product and the reference biological can be characterized to a high degree of confidence by physicochemical and in vitro techniques. Similar biologics are comparable to reference biologicals in all preclinical evaluations conducted. And pharmacokinetics and pharmacodynamic studies has demonstrated comparability of PD markers validated for clinical outcome and has preferentially been done in inpatient setting with safety measurement for adequate period. So a comprehensive post-marketing risk management plan has been presented that will gather additional safety data with special emphasis on gathering immunogenicity data is also very, very important. So friend, confirmatory clinical safety and efficacy study cannot be waived, especially for large molecular weight biologicals like monoclonal antibodies. So these are all important conditions we have kept in our guidance document when we can go for waiver of the clinical trial permission in India. Next one, friend. Then CTD format, they have to submit as per the CTD format. So what are the important principles adopted in CDNCO guidance document for industry? Post-market data for similar biological, in that they have to give what is the risk management plan, how many indicators they have made application, submission of periodic safety update report, pharmacovigilance plan, other adverse drug reaction reporting form as per the Schedule Y, then safety data on more than 200 patients within 2 years. So if safety information, if the more subjects are not involved during the clinical trial phase, then subject expert committee members can give the structured phase 4 study to carry out of the respective marked authorization given with the condition to submit the safety data information on at least 200 patients and completion within 2 years. So this is the targeted sample size we have given, and for the phase 3 clinical trials, at least 100 that we have elaborated the sample size. At least 100 minimum available patient shall be undertaken during that phase 3 clinical trial when they go for this biosimilar product clinical trial. So they are all important principles we have incorporated. So you can go through that, and it is publicable. It is public. It is a public document. It is available in our website also, and you can go through that, and you can have a view with respect to the manufacturing activities and clinical trial permissions. Next one, friend. So these are all various forms as per the Drug and Cosmetic Act and as per the city rules and 2019 published in the month of March. So these are all various applications with respect to those who want to go for import license. They have to make application form city 16 and approval will come in city 17. So form 30 for manufacturing license, testing, analysis, examination, that is form 29 NOC. These are all things you can go through that and you can remember and you can buy hard for your benefit when you want to go for application to CDSCO, that is National Regulatory Authority. Next one, friend. And revision. So in 2012 there was a need for, because what are the important points. So this is because why we have to go for revision. So they have mentioned in that. So need for regulations, need for guidelines. Biologics are complex proteins manufactured by ordinary technology and different from chemical drugs, requiring advanced development and evaluation process. Therefore, separate set of guidelines are needed. So this is consistent with the approach being taken by all drug regulatory authorities. So accordingly, we have modified in 2016. Next one, friend. So guidelines on biosimilars, key points published in the year 2012. If you see, guidelines were prepared, finalized, uploaded in a website of CDS in October 2012, elucidated the regulatory requirement for market authorization of similar biological products in India, prepared by DBT and CDS in a consultation with various stakeholders, experts from premier institutes like CDRA, Lucknow, PGMR, Chandigarh, IIT Delhi, NAN, Hyderabad, IIC Bangalore. So during the discussion of 2012 guidelines preparation, it was anticipated by the group of experts that there would be need for ongoing review and revisions from time to time. So due to that point, so we have revised in 2016, friends. Next one, please. So detailed guidelines on regulatory path, framework for comparability exercise for similar biological with reference product, guidance on specific requirements with respect to product characterization, including physicochemical parameters, preclinical evaluation, clinical evaluation, pharmacodynamics, safety, efficacy, immunogenicity, till the pharmacovigilance, till the post-marketing authorization is concerned. So there are all points added in 2012 guidelines, and what are the basic changes took place in 2016 that we can see. Next one, friend. So rationale for review that we have covered. Next one. So rationale for review, because the broad framework of guidelines remain unchanged. However, the revision seeks to enhance the scientific utility of this guideline to ensure the robust quality and clinical evaluation by explicitly defining. First time we have incorporated the definition of the similar biological product, identified the critical quality attributes for establishing similarity, specifying lower limits of minimum sample size for clinical evaluation that we have discussed. At least 100 minimum available patients should be, subjects shall be enrolled during the phase 3 clinical trial program. They are making phase 4 safety evaluations mandatory, specifying lower limits of minimum sample size for it. So at least not less than 200 patients and should be completed within 2 years. So that was the cutoff we have included in phase 4 clinical trial program. Next one, friend. So they're all okay. Next one. Key revisions. So if you see what are the key revisions and how we have focused with respect to technicalities involved, more detailed guidance and specific revisions are proposed in 3 distinct areas that are fundamental to the regulation of this class of complex products. One is approach to the quality comparability study. Second one is pre-approval clinical evaluation with respect to safety and efficacy. Third one is the post-marketing safety evaluation using phase 4 studies. So these are the three important things we have added in our existing 2016 guidelines. Next one, friend. Then there are all things. So quality comparability data. Specific section on this quality comparability study has been added that explicitly defines a class of quality attributes, critical quality attributes where the proposed similar biological has to be within the range that is established based on the reference biological product. So this establishes a framework for product similarity that can be applied across all approved similar biologics and is consistent with approach that EMEA, USFDA have taken. Next one. And quality comparability in this, in addition to critical quality attributes, the revised guidelines also define key quality attributes, KQAs, which define all other parameters that need to be well controlled to ensure the safety consistency of the product. So this approach to the classification of quality attributes establishes a solid foundation on which the remainder of the development program for a similar biological can be built. In future, it will facilitate the development of product specific guidelines that will help to establish the expectations for higher quality similar biological products. So we are not having as on date for specific product related, but in general we had detailed guidelines in similar line of ICH and USFDA and EMA guidelines for approval of biosimilars in India. Next one, friend. So because time is less, next I have to attend for the UK conference, so that is a problem. So you understand, and we are having more slides, and I will cover only important slides so that you can understand, and the rest you can email to me with respect to the queries if you have anything. Next one, friend. So post-marketing, here we have added phase 4 study. Here there has also been significant addition to the post-approval expectation of the phase 4 study in more than 200 available patients to create a minimum safety database of at least 300 patients treated in controlled setting for each approved similar biological product. So this is the sample size we have added in 2016 guidelines. Next one, friend. Then summary of key changes as per the proposed guidelines section wise. Introduction we have added, that is definition, similar biological product which is similar in terms of quality, safety, efficacy to an approved reference biological product based on comparability is called as biosimilar. Then background objectives, no changes. Applicable regulations, guidelines, no changes. Next one, friend. Scope, requirement of reference biological have been revised, and 2012 guideline, reference biological should have been licensed and marketed for at least 4 years in a ICS country, if not authorized in India. But in 2016 guideline, we have added reference biologicals should be approved or licensed in a ICS country, if not authorized in India. That we have added. So this is to be considered in light of full preclinical and clinical evaluation expected for a similar biological product. So this is a thing we have added in our 2016 guidelines, friends. Next one. And principles for development of similar biological. Selection of reference biological, no change. Manufacturing process, no change. Quality based consideration for similar biological, no change. And it is in line of 2012 guidelines only. Next one, friend. And here, quality comparability study now defines critical quality attributes, key quality attributes. We have defined critical quality attributes for similar biological has to be within the range that is established based on the reference biological. Key quality attributes for similar biological has to be well controlled to ensure the consistency of the product. Here, consistency is very, very important. Analytical test required for comprehensive quality comparability exercise of critical and key quality attributes are suggested. Next one, friend. Data requirement for preclinical studies, procedural simplification you have done, and post availability of toxicological study report, clinical trial application, toxicity study report to RCGM can be filed in parallel way. However, DCGI will issue clinical trial NOC only upon clearance from RCGM, but at least we have given the flexibility to have parallel submission so that they can have the less time and they can reduce the time for the approval process. Next one, friend. And data requirement for clinical trial application. Here, the 2012 guideline, no guidance on minimum phase 3 sample size was provided. So in 2016 guideline, we have added clear expectation of comparative safety and efficacy study with the explicit requirement of not less than 100 available subjects on the test arm. This sets a minimum bar on pre-approval data to establish the comparable efficacy. So first time we have incorporated in 2016 guideline so that people can take care and we can get the data with respect to the sample size. The protocol for post-marketing phase 4 open label single arm safety study in not less than 200 subjects on similar that already we have discussed. So this takes care of additional safety data to have the robust safety database. 100 plus 200, almost 300 people, 300 subjects we are covering. So that will facilitate and to establish the safety of the proposed similar biological products approval by the regulatory agencies. So this is a thing as per the recommendations of, as per the suggestions of experts, we have added and took the decision and published that CDSO and DBT guidelines in 2016. Next one, friend. So data requirement for market authorization application, post-marketing, no change. So rest of the important points are almost all in similar to 2012 guidelines. Next one. Application forms, archival data, applicability. All these are because mentioned in a detailed way with respect to the comparison. Next one. So friends, so these are all important things. So whatever I gave the important principles, key quality attributes, critical quality attributes, the reference biological products and the similar biological products, and what are the key changes took place with respect to 2012 and 2016 guidelines. So everyone can understand. So time is very less, so you understand. So next time definitely I will take 1 hour detailed presentation, but due to my preoccupied with the UK and India, the important webinar it is going to start at 2:30 on medical devices and in vitro diagnostics. So I could not be able to spend more time with you, but these are all some of the important principles as per the request of Ruchika Daveji, and I would like to congratulate all the organizing committee members for taking this important webinar for the benefit of all student fraternity and other stakeholders also. So friend, there are innumerable qualities that a person can have to live with respect to the quality life. So the person should have the humility, kindness and patience and strong work ethics culture and enthusiasm to learn and constantly improve upon what we call the good life. So therefore, life is like a game, juggling some 5 balls in the air. They are like work, family, health, friends and spirit. So you will soon understand that work is like a rubber ball. If you drop it, it will bounce back, but the other 4 balls like family, health, friends and spirit are made up of glass. So they are very, very danger, and it is very, once it is dropped, then it will be irrevocably scuffed, damaged, marked, naked or even shattered. So they will never be the same. So you must understand that, strive for it, work efficiently, work hard during your office and study hours, and leave on time, give required time to your family, friends and parents, have proper rest. So value has a value only if its value is value. So thank you very much for giving me the opportunity to share my thoughts for the betterment of all the participants, invited guests, and delegates attending across the country with respect to this today's webinar being organized by this Patkar College, Mumbai. Thank you very much. Hello? Unmute చేయండి.
Thank you so much, sir, for this informative talk. So, with your permission, can we move to the question and answer session? We have just one question, sir. The question is that if the drug is developed in India for Indians, so is it necessary to take the approval from other countries?
Equal BOs in their population. So they may go for phase 3 clinical trial. This thing they will ask to submit the data for at least phase 3 if drug is approved in India. If they want to go for other countries market authorization, then they have to follow their respective FDA requirements. But in India, if molecule is not approved in India, if molecule is approved outside of India, if they want to go for market authorization or import authorization in India, then we will request them to submit the phase 3 clinical trial data.
Okay, thank you very much, sir. Thank you, sir, for taking out time from your busy schedule and delivering us such an informative talk on regulatory requirements for approval of biosimilar in India. Thank you, sir.
Thank you.
After this wonderful session, we'll move ahead with the last session of the day. I would like to introduce our next speaker, Dr. Sanjeev Gupta. Sanjeev Gupta has obtained PhD in molecular biology, cell line engineering, and MSc in applied microbiology and biotechnology. At present, working as senior general manager and head, Advanced Biotech Lab, R&D Biosimilar in IPCA Laboratories, Mumbai. Sanjeev Gupta has established a new world-class state-of-the-art biotech R&D facility for the development of MAB biosimilars. He is also actively involved in establishing modern, lean, and multi-product flexible single use of biomanufacturing facility for biosimilars clinical trials and its commercial launch. He carries around 21 years of industrial experience and has been working since 2000 on the development of biosimilars, including monoclonal antibodies. His core expertise lies into molecular biology, cell lines, upstream process development, bioanalytical, as well as tech transfer to GMP for clinical and commercial batches. Till date, he developed and contributed for over 12 recombinant molecules, of which 8 biosimilar products has been already launched in the Indian market, and the rest are in the development or clinical phase and are expected to be launched in near future. During his professional tenure, he worked at various positions in reputed Indian biopharmaceutical industries such as Zydus, Ahmedabad, Wokat, Aurangabad, Panacea, New Delhi, Intas, Ahmedabad, and at present with IPCA Laboratories, Mumbai. He delivered talks on biosimilars in various national and international conferences organized by IBC, Terrapin, Biotrain, IMPA, IMAPAC, CPHI, Biopharma, Informa, UBM. He has also published several research articles, book chapters, magazines, and patents on the topic of biosimilar development. In his honor, it is honor to have such an eminent personality with us here today. So let us hear from Dr. Sanjeev Gupta sir on current challenges in biosimilar development and approvals. I would request sir to take over the session. Thank you.
Thank you very much, Ms. Sweta. I am at home now. I'm not in the office, so I am attending this session from home only. Thank you very much for your kind introduction, and I would like to thank organizing committee also for giving me such a wonderful opportunity, especially Ms. Ruchita Dalvi, and for good coordination, Mr. Pushkar. Thank you very much for organizing this wonderful session. So, as Ms. Sweta told that I'm going to cover the current challenges in biosimilar development and approvals. So it was really very nice session before my talk. Dr. Ramakrishnan has covered most of the things about the approvals, regulatory approvals, which are required for the biosimilar development. So I'm not going to touch up more upon on those aspects, but I'll just give you the brief idea what are the things required for the biosimilar development in terms of regulatory approvals. So, am I audible? I hope I am audible clearly. Hello?
Yes, sir, you are audible.
Okay. So, in today's discussion, I am going to cover this topic. I will give the introduction about the IPCA because some of you may not be knowing who we are, what IPCA does. IPCA is a pharma company, but we venture into biopharma also. And I will also touch upon the biologics and biosimilar. What is the difference and what is the opportunities for the Indian biosimilar and the students who are doing MTech, MSc, or PhD? What are the opportunity for those students who are into this area like biotechnology or microbiology? So I'll touch upon those things. Then the current challenges in biosimilar development. When we develop the biosimilar, what are the challenges we face? Indian industries or any industries who develop biosimilar, what are the major challenges and how we cope up with these challenges to be able to launch the product in the Indian market or rest of the world. Then I will also touch upon the biosimilar development path, the flow path, how do we develop the biosimilar, and some of the technical aspects I will touch upon like cell engineering, which is very fascinating because everything starts from the clone. We have to engineer the cell and develop the process. Then ultimately we develop the biosimilar. And I'll touch upon the concluding remarks. So before going into the technical detail, I'll just give you the brief idea about my company, which I represent. So IPCA is a Mumbai-based company, and this is incorporated in 1979. And if you see the total sales, it has been about $700 million, about 5,000 crore company. If you see the number of employees we have 16,000 employees across India. And our business model is fully integrated pharma company producing branded generic formulation, APIs, and intermediates. But very recently, we ventured into biopharma business in 2015. We established a state-of-the-art R&D center in western part of Mumbai, that is in Kandivali West. We are laced with modern. Our labs are well equipped with the modern equipments and high throughput devices, which are required for the biotechnology. Because in biotechnology, especially biosimilar, biologics, or gene therapy, high throughput devices are required to be able to produce a good clone or good processes. So we are laced with all these equipments. I'm giving this background because this is very important to understand the requirement for the biologics and biosimilar development. So what kind of facility we have, what kind of infrastructure which we have, and we are able to deliver the clone, clinic, all these things. So if we talk about IPCA, we are capable of in-house development of clone as well as processes. So we do not in-license clone, or we do not in-license any technology to from third party like USA. Most of the companies in India, we in-license. So I'm not saying that this is not a good strategy. This is a good strategy, but our philosophy is to develop clone processes in-house, just like self-reliance, you can say. So we are capable of developing clone, and that clone and process goes up to the clinical. And as far as systems are concerned, we are working only on mammalian system. We do not have E. coli system. We do not have yeast or fungal system, and very much focused on monoclonal antibody. We call it MAPS. So therapeutic MAPS, we call it magic bullet as well. If you see our scientific staff, we have 50 to 60 scientists working on the biosimilar area. And single use, we are also developing single use biomanufacturing facility. I'll just touch upon this, what is single use biomanufacturing facility, which will become functional by Q2 2023. So this is R&D functional structure. This will give you the brief idea about the functional structure which we have, and also the process flow. How do we develop the molecule, whether it is biologics or biosimilar? So let's start with first step, which is molecular biology and cell line development. So for any biologics or any biosimilar, the clone, the cell line is the raw material from where we start everything. So as I rightly mentioned, we develop clone and processes in-house. So we have facility wherein we do the molecular biology work. We do the genetic manipulation in E. coli. Then after E. coli work, we move to the cell line, that is mammalian cell line, and we develop the cell line. This is extensive process. This takes around 9 to 12 months time. In later part of my talk, I will discuss about the cell line development. How do we develop the cell line? How do we develop the processes which are economical and competent to be able to survive in the market? Once clone is developed, we move to the second step. You can see here that is upstream process. Upstream process is nothing but cell culture, or you can say fermentation, wherein we optimize the process. A lot of tweakings are required, like media, feed, process development, then temperature shift, all these things, which I will describe you later part of my talk. Then we move to the downstream process, which is purification. So whatever molecule we develop in fermentation or upstream, we have to purify, because ultimately it will go to the human body. It will be injected to human body. So it should be very, very pure. After that, after purification, we go for the formulation and stability studies. This is again, all these steps are regulatory requirement. And whatever we do in industry, we follow the regulatory norms. Right? As Dr. Ramakrishnan mentioned about 2016 guideline. So we follow those 2016 guidelines, as well as in addition to that, there are some certain other guidelines like ICS guidelines, WHO guidelines, FDA guidelines. All these guidelines we follow to develop a molecule. It's a very cumbersome, complex, time-consuming, and challenging process. And you can see here, the middle part of this workflow, that is analytical and biopsy, which is very, very important. So samples from every department, reaches to analytical and biopsy department, and they verify your molecule. They verify whatever you are producing, whatever we are producing, that is up to the mark and comparable with the innovator, that is biologics. So this function is very, very important. After this, we move to the preclinical study, that is animal study, and wherein regulatory department and QA department plays major role. Though these departments are very much involved right from the beginning, but when we reach to the preclinical stage, they play major role. Then followed by phase 1, human phase 1, phase 2 trial, phase 3 trial, sorry. So that was the workflow, the functions which we have at IPCA. Any company who are developing the molecule biosimilar biologics, they would be having this kind of workflow and the functions. So these are the molecules which we have in our pipeline. And we could not disclose the name of the molecule, but we are working on 5 monoclonal antibodies. And these antibodies are used for the cancer therapy as well as inflammatory diseases like rheumatoid arthritis, colitis, psoriasis, spondylitis. All these molecules are being developed. In fact, IPCA is well known for the pain management, like rheumatoid arthritis or any other pain management. And we have also marketed 3 biosimilar products. Because we started 2015 onwards, we are not able to launch our own products. So we are doing P2P business. We have 3 molecules in the market. Now I'll move to the next step, that is biologics and biosimilars. I will describe here what is biologics and what is biosimilar, what is the difference, and what is the small molecule. If you see this slide, this describes that what is the difference between a small molecule and biologics, which is very, very complex. You can see here it is written that biologics are larger and structurally more complex than small molecule. If you see at the left part of the slide, this is a small molecule, which is acetyl salicylic acid. The atoms, if you see the number of atoms, it has got only 21 atoms. And the molecular weight is 180 Dalton. Whereas if you move to the biologics, this side, right side of the slides, the number of atoms are 25,000. And the molecular weight is 150 kDa, which is almost 1,000 times greater than a small molecule, which I am quoting example here. So you can see that how complex it is structure wise, and a lot of post translational modification occurs in a monoclonal antibody, which I will describe later part of the presentation. Now here you can see what is biologics and what is we can differentiate biologics versus biosimilar. Biologics are complex recombinant molecule developed by innovators. For any molecule which is developed by first company or innovator company, we call it biologics. And it is developed using living organisms like bacterial system, mammalian system, yeast cells. All these systems, all these hosts are used for the development of biologics. Now they develop as per their country guideline, like EMA, US FDA. They launch the product, they launch in other part of the country. Now the other companies like biosimilar companies, they make replica of that particular molecule, and that's why we call it biosimilar. In case of small molecule, we call it generic molecule, but in case of biologics, we call it biosimilar. Similar to the biologics, we call it biosimilars. So what is biosimilar? Biosimilars are highly similar to biologics, but it is not identical. In case of small molecule, we can say that it is identical. In fact, it is identical. But in biosimilar, it is impossible to make the identical molecule, because we deal with the biological system. So a lot of post-translational modification, process optimization, that affects the critical quality attributes or maybe key quality attributes of the molecules. We cannot say that in case of biosimilar, we make identical molecule. We make similar, and we call it, when we qualify a molecule is highly similar, then regulator approves that molecule for biosimilarity as a biosimilar, and we are able to launch the product in the market. The second thing is no clinically meaningful differences in safety, purity, and potency. So whatever molecule we develop, there should not be meaningful differences in safety. We don't have to compromise with the safety, purity, and potency of the molecule. So I think Dr. Ramakrishnan has covered all these things, but I thought that I'll just touch upon these things. Now the question arises: why biologics? Why biosimilar? Why Indian companies or other pharma companies should venture into biologics or biosimilar? In India, several companies are there. I think 2-3 companies, not several. 2-3 companies are there who are working on biologics. But more than 20-30 companies are there in India who are working on biosimilars. So why biologics? These biologics provide unmet therapy needs. And also, if you see, compared to the pharma drug, it shows less side effect. That's why it is preferred. Biologics and biosimilars are preferred, but at the same time, the cost of therapy is very, very high compared to a small molecule. So if you see the number of blockbuster, out of 15 blockbuster in the market, which includes pharma, biopharma, 10 molecules are biologics. You can see it is really gearing up and taking place and taking over the small molecule. If you see the number of approvals which US FDA had given, I have data up to 2018 only. So every year, if you see 2015 onwards, 45, 22, 46, 47, which includes total number of drugs. Out of these number of drugs, if you see the biologics, 13, 14, 22, 18 every year. It's really very, very good number, I would say, because biologics takes long time to get into the market, come into the market. Projected sales, if you see forecast of biologics, in 2016 it was $210 billion, but in 2024 it is projected that it will become, the market size will become $480 billion, which is very, very good. and this is growing with at the rate of 10 to 11 CAGR every year. This slide gives you just glimpse of the top 15 pharma products. If you see this was published in 2019. Out of, I already explained that out of 15 molecules, 10 molecules are biologics. And these are the biologics you can see, biologics plus, pharma molecule. Humira, this is topmost $20 billion molecule. Then Keytruda, these are again biologics. All these, if you see Eylea, these are the biologics. So it has great potential. Why I'm showing this slide to this forum? Because first of all, we should learn and we should be knowing what is the importance, what is the market size. Why should we be working on the biologics or biosimilar? Why companies are working on the? This is the reason. The first reason is the business, good potential, and the second reason is therapy for the human welfare. So that's why most of the companies, pharma companies, are slowly venturing into biopharma because it huge potential. It offers huge potential in terms of market size and the benefit. So if you see number of molecules which are being developed, one molecule, if you are developing one molecule, biologics, biosimilar, it takes around 7 to 10 years time to be able to launch in the market. So you can imagine more than 400, 500 molecules are being developed. Phase 1, almost 285 molecules. 200 molecules corresponds to cancer therapy. That means that 200 molecules will be developed for the cancer therapy and 85 for non-cancer. This is in phase 1. Then similarly, phase 2, 225 molecules. Then late stage of the study means phase 3, and they are ready to launch around 60 molecules. So you can imagine how many molecules are being developed for the cancer, non-cancer therapy. So we have huge potential, a huge opportunity for the companies as well as students who are doing graduation or PhD in this area. Now, if you see the biosimilar market, earlier I discussed about the biologics market. Now I'll discuss here the biosimilar market. If you see the biosimilar market, in 2020, it has been 5.5 billion. It's a global sale, but it is predicted that by 2030, it will be $250 billion, which is huge. And if you see the Indian market, in 2018, it was more than $1 billion, but in 2023, it is predicted that it will be $35 billion, which is huge. So that's why most of the Indian pharma companies are already ventured into biopharma, and they are working on biosimilar. What is the opportunity? Why do we need to develop the biosimilar? Because this patent, that for any biologics, any new molecule, any new platform, there will be patent. But these patents are going to be expired, means already expired, worth of $75 billion in 2020. That's why whole of companies are working in this area. So this is about the potential opportunity market, but at the same time, we have a lot of challenges because so many companies are working on biosimilar. Almost, you can say that on the same molecule, more than 100 companies are working. So how to compete in this, competitive market? This is very, very difficult to survive and sustain in this biosimilar market. So rising competition in the market is the challenge, as I said. And access to the right technology, biotechnology, the platform technology, the process, the clone is very, very important. If you are able to choose a good clone, a good process, a good technology, you may survive or you may sustain. I will describe you later part of the discussion. And if you are not, then you may have to drop in between, or maybe after 5 years or 7 years down the line, you will realize that this molecule is not potential and let's drop it out. So when we select a molecule, because in biosimilar biologics, there are more than, I think, 60, 70 molecules are there, which we can develop as a biosimilar. So we have to be very cautious when we select the molecule. We have to focus on the number of patients, the market size, and the competition among the industry, and then we select the molecule. And we have to also forecast what will happen 10 years down the line, what will happen 15 years down the line, because this takes 7 to 10 years time to get delivered into the market. The third challenge is regulatory approvals, which is very, very challenging and very, very time consuming. At each step of the development, we have to approach RCGM, either RCGM or DCGI, and get the approval to move to the next step. If they have some doubt, if regulator have some doubt, or if they have some, queries, we will have to answer those queries, and sometime we may have to repeat the experiment also. So it's a time consuming process and challenging process. And fourth step, which is very, very important, and every company is facing this issue, that is price erosion. In India, if I take example of one molecule, that is rituximab, I think more than 10 companies are, they have launched this molecule. So there are price, significant price erosion from 100% to 70% if you compare with other biosimilar or innovator products. So today, if you are launching a product, let's say with the cost of 10,000 rupees, tomorrow after 5 years or 7 years, you should be ready to sell the same molecule with 3,000 rupees or 2,000 rupees, because after 5 or 7 years there will be a lot of competition, and with the advent of new technology, novel thing, and the new companies who are developing the same molecule might get better yield and good quality of the products, and they might reduce the price significantly. So every company should be ready. We say that every molecule has 10 to 15 years of life. So, but to address these challenges, what are the approach? How can we sustain in the market? Lowering developmental cost via innovative approach is the key for sustenance in the high competitive biosimilar area. So we have to work on, in fact, every company is working on that. How can we lower the cost of the development? If we have good clone, good process, if you are increasing your yield, if you are enhancing your quality of the molecules, you will be able to reduce the cost significantly, and we can sustain in the market. If not, then no. So these are the things which we have to keep in our mind for the students, also for the PhD scholars when they venture into this biosimilar business. These are the key things which we have to think, and we have to be innovative to compete in the market. Whether you are doing, a molecular biology, a cell culture, a downstream process, formulation, each and every area we have to think that how can we reduce the cost by innovative approach. Now, this is just to give you a glimpse. This was published in 2018. That how many molecules are being developed in each country. If you see China, I think it was 270 molecule in 2017, 18, but now it would have crossed 300 molecules. Likewise in India, 260 molecules. So many companies are working on biosimilar. United States, 200 molecules. South Korea, Russia, Switzerland. So we have to do the market assessment and see that which company is working on what, and based on that we select the molecule. This is our approach. This is every company does that. So I'll move to the technical part now. This the whole before this talk, before this slide, whatever we discussed about the market potential, the challenges, and the opportunity in biosimilar area. Now I will touch upon the technical part of the biosimilar. So as I stated, we are working on monoclonal antibodies. Everybody knows that what is monoclonal antibody, but I wanted to just refresh the structure of the monoclonal antibody. If you see, this is Y-shaped structure, and if you see in these arms, there are several modification: pyroglutamine, deamidation, methionine oxidation, glycation, mannose, and this is the glycosylation, galactosylation, and glycosylation, which is very, very important for the activity of the molecules. This is all these modifications which are stated here. This is post-translational modification. And when we develop the molecule, whether it is clone, a process, upstream process, downstream process, formulation, we have to ensure that our molecule is intact and the quality is matching with the biologics or innovator molecules. If you see, if you combine all these modifications, how many modifications may occur in a single molecule? It's 285 million. Can you imagine? This is published by FDA. So that's why we say that we say that the developing biosimilar biologics is very challenging and very complex. So this will just give you idea. Now this is the process flow. I think most of you are aware of the process. How does it goes? As I I discussed earlier also. It starts with molecular biology, cell lines. So we design the gene and then we transfer it to the cells. Then we develop the clone. Once clone is developed, this moves to the upstream process. So you can see that this is seed generation, small scale, then little bigger scale, we call it N-1. Then it goes to the bioreactor, that is cell culture upstream process. Here you can see one vial. So when we develop the clone, we prepare the cell bank and we start from here. We thaw the vial and then we expand the cells and then it goes to the upstream process. When we reach this process, this cell line development process takes around 9 to 12 months time. Once cell line is developed, we bank the cells, we prepare the master cell bank, our working cell bank, then we start our process from here. So cell line revival and reaching up to here takes around 15 to 20 days time. That solely depends on your process and clones and cell line behavior. Then we do the process development upstream process, which takes around 14 to 18 days time. That again depends on your process. After this, once upstream process is complete, we move to the downstream process. So we have multiple chromatography step, protein A escape, then protein A purification, then polishing step like cation exchange chromatography, anion exchange chromatography. All these steps are involved, which purify the protein and removes the impurity which comes from the upstream process or the cell culture process. And after this, we make the molecule, we formulate it, and that goes for the injection. In one slide, we can summarize everything, but it takes around 7 to 9 years time. And every step, we need to take the regulatory approval. If the regulator says, yes, this is okay, then go ahead. Now, this is the regulatory step. This is nutshell. In nutshell, I have just described here the regulatory landscape, which is for the biosimilar development in India. So if you see, we have segregated here two parts. That is, first case up to here, that is research and development, and here the manufacturing. So we do develop clone and process in R&D, and then we take the permission from RCGM and local FDA for several activities like carrying out research, the NOC from the regulator. Then after the process development, we move to the preclinical studies, that is animal study. So we approach RCGM, regulatory body, and we show the data, and based on the data, they give us approval for the preclinical trial, that is animal tox study. And once we get the animal tox study done and we get the approval, NOC from RCGM forward our application to DCGI. And when we reach to DCGI stage, let's say phase 1 and phase 3 trial, human clinical trial, we have to manufacture our molecule in CGMP manufacturing facility. We cannot develop a molecule, we cannot manufacture our molecule in research and development, because in research and development, the environment is different. We do not use for the human use. But the moment we start using for the human clinical trial or maybe launch of the product, we do the tech transfer from R&D to manufacturing. So this is the entire process. And after phase 1, phase 3 trial, as Dr. Ramakrishnan was describing, we need to do the phase 1, phase 3 trial for 200 to 300 patients. It takes around one year time, 1.5 year time. That depends on the molecule, the mechanism of action, as well as the regulatory process. Sometimes it takes 2 to 3 years time for a given molecule. And one thing I would like to emphasize here, that in biosimilar, we do not do phase 2 trial. Phase 2 trial, phase 1, phase 2, and phase 3 trial is being done for the biologics. So that's where we get the waiver and we do phase 1 and phase 3 trial to be able to get the approval for the launch of the product. Not only India, if you see the European guideline, even USA guideline, for the biosimilar, phase 2 trial is not required. So this is the regulatory landscape which we follow for the development of biosimilar. Now, I'll touch upon the cell line and upstream process, which is the key thing, key raw material, I would say, for any biosimilar development or any biologics development. So I'll just give you a brief idea. How do we develop the clone? How do we develop the process? And how do we develop the entire biosimilar molecule? So in cell line development, there are two approaches: conventional approaches and novel approach. As I mentioned, we have to always be innovative and working on the innovation to develop a good cell line, high producer cell line, which can give us good quality of the product. So there are two processes here approaches: the conventional approaches as well as novel approaches. So I describe here what do we expect from a clone? When we develop the clone, what are the expectations? The clone should be high expressive. It should be giving very good yield so that we can compete in the market. It should be commercially viable in the market. Suppose, for example, I have a clone which is giving 1 gram per liter expression. Another company is having clone which gives 3 gram per liter expression, or maybe 5 gram per liter expression. We are nowhere. I am nowhere. To produce the same amount of the protein, quantity of the protein, we might have to scale up our process at 5,000 liter scale. The cost of development goes high. But the company who is having 5 gram per liter title clone, they may have to produce their protein in 1,000 liter scale. At 1,000 liter bioreactor scale, the cost of development goes down, goes low, maybe 3 to 5 times. So that's why high expression is desired. We should get very good expression when we develop a clone. Stability. The clone should be stable because we play around with the genetic manipulation. Your gene of interest goes and integrated into the CHO genome or any other host cell genome like yeast genome. But after development of the clone, we see the stability of the molecule. It should not lose the gene copy number. If it is losing, that may create the heterogeneity of the molecule. We may not expect the uniformity of the molecule in terms of expression as well as quality of the protein. Clonality, which is very, very important. When we develop the clone, we have to ensure that our clone has been generated from a single cell. The regulator asks to provide the data. I'll show you some of the slide. How do we ensure that the clone which we develop that is generated from a single cell? Like for example, you would have done the transformation, E. coli transformation. When we do the transformation, we get the isolated colonies on the plate and we pick up single cell, single clone, single colony, and then transfer into the tube or shake flask for the growth and the further study. Similarly, in case of in case of mammalian system, we do not have flexibility to plate on the agar plate and then get the clone. But we have to ensure that there are tools available which I will describe, which can help you in getting the single cell clone from a single well, and you can show the regulator that the clone which has been derived that is from single cell. Scalability. The next part is scalability. When we develop the clone, when we do the study, we do at small scale, let's say shake flask scale or ember. But it should be scalable at higher scale because we scale up the clone from small scale to let's say 1,000 liter scale, 5,000 liter scale. So we study all these parameters whether the clone can be scaled up to higher scale or not. Then we select the clone. Good quality, which is very, very important. That is biosimilarity. Our regulator emphasize on this biosimilarity, especially the key quality attribute and critical quality attributes of the molecule. Until unless we are meeting the requirement, we cannot qualify for the biosimilarity. So these are the desire when we develop. We desire when we develop the clone, and we think that if the clone qualifies all these criteria, that can be considered as a commercially viable and we can scale up the process. Now, the second question is how to select the host. Like we have E. coli, we have prokaryotic E. coli, then we have yeast, then we have mammalian. All these platforms are available. How to select the platform? When you have large protein, we have to choose eukaryotes. The large protein like monocular antibody cannot be expressed in E. coli because it has its own limitation. When you have small protein, when we have small protein, then we can choose prokaryotes. When your protein is glycosylated protein, we have to choose mammalian system because post-translation modification occurs in mammalian system only. When you do not require post-translation modification, you require high yield, low cost, then choose E. coli. So these are the criteria when we select the platform. But monoclonal antibodies, for monoclonal antibody, we have to select the mammalian system like CHO expression system. Without mammalian system, we cannot develop the map. So the cloning initiation, when we start the clone, what are the criteria? What are the things which we need to see? Like suitable host. There are so many hosts. Even within mammalian system, so many hosts are available. You can see CHO-K1 strain, CHO-S, CHO-M, CHO-DHFR, CHO-DH, all these strains are available. Mouse myeloma cell line, SP2, NSO, human cell line. So we have to study all these things, and based on certain criteria, we select a suitable host, and then we move for the clone development. We have several platforms like DHFR platform, GS platform. I could not describe all these things with limited time, but maybe you can study, you can see what is DHFR platform, what is GS platform, antibiotic resistance platform. So we select suitable host, we select suitable vector, we put our gene of interest into the vector, we characterize it, and then we transfer it into these hosts. And then we develop the clone or cell line. So what happens in conventional approach? In conventional approach, what we do is we select the vector and we select the particular gene, we put together, and then we start the expression, we develop the clone. You can see here one of the pictorial, that is DHFR gene. Why do we put DHFR here? DHFR helps in increasing the gene copy number when we select the clone in presence of MTX. So that's the purpose of selecting the platform. When you transcript any gene, your cell may have one cell or one gene copy number, two gene copy number, five gene copy number. But to be able to increase the protein expression or to increase the protein expression, we have to amplify the gene. So there are several steps involved which helps in amplifying the gene. We increase the MTX from low concentration to high concentration. That's how genes are amplified inside the genome. So this is the conventional approach. But what are the major challenges with the conventional approach? We get low expression. We need to put more efforts, and we face challenges in clones instability. Because when we develop, when we have more gene copy number, it may lead to the gene copy loss and instability of the clones. So these are the conventional approach and challenges which we face. I think I have already described this. I will move to the next part. But what is the current trends? What is the novel approach for the biosimilar development? I described some of the things. So many platforms are there people are using nowadays. Those are the novel platform, but I will touch upon some of the novel platform. You might be aware of those platforms. Before that, I just would like to here mention that there are two parts in genome, whether it is human genome or CHO genome. You might have studied all these things: heterochromatin region and euchromatin region. Heterochromatin region is condensed region, and euchromatin region is extended. Euchromatin is transcriptionally active site and active, and this is heterochromatin transcriptionally inactive site. This is less methylated, this is high methylated. So our aim is to introduce our gene of interest at euchromatin site so that our transcription and protein expression does not stop and clone shows high stability and uniformity of the molecules. So with this concept, if you see the conventional approach, we put the gene randomly. It goes. Sometimes it integrates into the heterochromatin region. Sometimes it integrates into the euchromatin region. But when you do the site-specific integration in particular genome, you are aware and you are sure that your gene of interest is going and that is integrating into the transcriptionally active site. So this is the novel approach people have started, and we are also working on this novel approach. So this is cell line engineering tools. You might have heard these tools, like these are the gene editing tools. You can edit the gene based on your requirement. If you want to knock out certain gene from the genome, you can knock out. You target. You use these three tools like CRISPR-Cas9, TALENS, and gene fingers, and you can knock out your gene. Or if you want to target certain site to overexpress your gene of interest, you can knock in the genes. So these are the tools I think in India, I think dozen of the companies have started working on it. We are also working on these tools to, target certain specific site for the knocking out of the gene or knocking in the gene so that we can get, good expression of the protein as well as good quality of the protein. So this is the tools. I think most of the research organizations have also started working on this, and this is the tool which is also used for the gene therapy. If you want to target certain gene in gene therapy, you can use CRISPR-Cas9 or TALENs or gene fingers to target certain gene in case of gene therapy. This is just to show you that how can we target the genome. Here you can see that this is CHO genome, and we can target any site in the CHO genome. We can have either multiple, single integration at single site or multiple gene in single site at single site or the multiple site. So we can design the strategy and we can target. And I'm happy to share with you that this paper was published by me in 2015 about the gene editing tools and how can we target the gene, how can we increase the expression and quality of the protein. So these are the application of gene editing tools. Like I already mentioned that gene knockout, gene knock-in, gene identification. Certain examples are given here, like carbon metabolism in cell line, lateral metabolism for the knockdown of the knocking out of certain genes. It can be used for overexpression of certain gene. It can be used, and as I mentioned, it is also used for the gene therapy. So this is the tools which we can use. This is called the recent tools, the novel approach, which can give you good clone and good quality of the protein. Now, I will describe the cell engineering workflow, how it goes. We develop the vector and then we transfer it into CHO cell line. Then we transfer into T25, T75 class, then it goes to the safe class. So what we do is we generate the stable pool. So we select the pool which contains non-producer clone, low producer clone, high producer, mediocre clone, and we assess the quality of the protein. So right from the beginning, when we start, developing the pool itself, without clone development, before clone development, we start assessing the quality of the molecules, whether we are getting good quality of the product or not. So this is the approach. Once you have a selected pool may have different kind of clone. Now you have to segregate them. So we use several tools. We can do single cell dilution cloning using manual method as well, but it's erroneous. So we use high end equipment like fax, and then we have cell imaging system. This tool is used for the transfection. Then once we generate the pool, we go for the fax for the single cell cloning in a 380 forward plate. Then we capture the image. For each and every well, we capture the image and we ensure that the well contains single cell. If the clone is derived from 2 cells or 3 cells, we disqualify and we reject the clone. Because regulator will not approve that, regulator will not accept. Also, that may lead to the heterogeneity of the molecule. Because if you have 2-3 different populations, you might get different kind of quality of the molecule. If you have single population, single cell, you will get the uniform quality of the molecule. So after this, we go for the 1,000 to 2,000 clone screening. After that, from 1,000 to 2,000, we zoom down to 10, 20 clones. We do the assessment, title, quality. You can see the tools. This is Amber, which is a small scale, bioreactor, which is 10 ml bioreactor. You can use this Amber tool for the process optimization for clone selection. This is again high throughput device. And I am happy to share with you that we are the first company who bought this equipment, Amber system, in India. Now, I think more than a dozen companies having this system. And this tool is very important for the clone screening as well as process development at a small scale. After this, once we select the clone, top clone, you can see here, I just wanted to give you example. This is the image of, if you see here, this is 380 voltage plate. So we put the clone here using the fax equipment. The volume is 40 microliter. Then we go for the cell imaging. We capture the image for each and every well. This is one of the example, one well. If you see day 0, here you can see one clone, one cell is there. we monitor, all these wells to select a clone which is derived from a single cell. Now you move to next step, day one. If you see day one, is kill one cell. Now day two, now cell has divided into four. Now cells are growing, and now we know that in this case we know that this four cells has been created from a single cell only. Now you move to the day five. Now cells are making the colony. These cells are spreading. Now these are making the colonies. Now go to day 8. So the example which I am showing here, we have to capture the plate. We have to remove the plate from CO2 incubator and capture the image in the cell imaging system, and then assess each and every well. And then we record it. A US FDA regulator, or maybe EMA regulator, or Indian regulator, they ask for this data. Whatever clones you have developed, you have to show that this has been derived from a single cell. You see day 12. So from single cell, now it is making the colony. Day 13. This is the process which we follow. Once it makes the colony, here you can see day 13, we lift the colony and we transfer to the higher scale, let's say 100 microliter or maybe 200 microliter, and then we scale up the clone. So this is the clone selection criteria. These are the clone selection criteria. We do all these studies for the clone selection, and then we move for the next step. Like we see the cell growth behavior, titer, QP, genetic stability, which I already described, which is very, very important. Process feasibility, clonality and purity, and product quality. At the same time, we see the potency and bioactivity of the molecule. If it is disqualified, we disqualify the clone, and we select the other clone. This is the flexibility we have. If we develop clone in-house, we have a lot of flexibility. If we get the clone or process from third party, we do not have flexibility. We get only one clone, and we are solely dependent on the third party. So that's why IPCA is working on this philosophy, self-reliance. We develop our own clone, we develop our own processes. Now, after this, the next step is process development. So process development that combines cell engineering process, the media field optimization process parameter. We combine all these parameters to be able to get high yield and good quality of the molecules. And we see here expression as well as post-translational modification of the molecule. And we expect high yield and good quality of the molecule. So with the interest of time, I will not describe much, but I will just touch upon the processes which we follow. So this is the screening workflow. Once we get the clone, we have battery of media and feed supplement. This is just a live cells. As you can consider, you have 10 babies, and their liking, disliking are different. They may like they may like different things. So likewise, here in case of clones also, we have to screen them with different media, feed, supplement, and then we assess their growth pattern, their quality, their titer, expression, and then zoom down the processes. We assess the quality and titer, and then we select final clone which goes to the ember, which is small scale. From ember, we move to the 5-liter scale bioreactor. So this is the process which we follow. This might take 6 to 9 months time. Depends on the clone's behavior and the molecule's nature. So this is the analytical parameter for any monoclonal antibody. These are the key quality attributes, that is KQA, and critical quality attribute, that is CQA, which we develop, which we analyze. At each and every step, we define what are the things which we need to analyze. So we have mass spectroscopy, we have SPLC, we have UPLCs. So we do the primary structure assessment, secondary and tertiary structure assessment, size of the molecules, charge profile of the molecules, glycosylation of the molecule. Then we also conduct the stability studies like force, degradation, whether our molecule is stable or not. Then we do the biological characterization like ADCC activity, CDC, then binding. All these assessment is being done. After this assessment, we compare with the innovator, and then we say that our molecule can be qualified as a biosimilar. So this is the cell culture process parameter. When we develop any process parameter, we optimize certain parameters like physical parameter. We optimize temperature, pressure, flow rate, shear force, education, and also biochemical parameters, DO, that is dissolved oxygen, carbon dioxide level, lactate, all these biochemical parameters. We look into all these parameters because these parameters affect the product title, yield, and quality of the molecules. So your upstream process, the clone and upstream process defines the fate of any biosimilar molecules. So we have to very much focus in this area, this particular field, and then develop a robust process which can give you consistent result and consistent product quality, and also we can be scaled up to larger scale when we go for the manufacturing. So this is the MAC development. This is summary of the development process which I described just now. We start with the MAP clone, then we go for the amber or shake flask, and then we go for the 5-liter scale. So we scale up the process, scale up the bioreactor size slowly, gradually. It's not like that from here amber we can go to 50 liter or maybe 2,000-liter scale. There are 5 to 10 times scale up, criteria. So we go from stage 1 to stage 2, that is 5-liter, from stage 2 to stage 3, that is 50-liter scale. From here, we go for a preclinical stage and get transferred to manufacturing. So we call it pilot scale. We develop this in R&D, and then we move to the manufacturing. So for manufacturing, we have several criteria. You can go for the phase wise, depends on the market demands, 200-liter scale, 500-liter scale. This is our strategy. We are following this strategy. And later on, when market demands increases, you can scale up your process up to 2,000-liter scale or maybe 5,000-liter, bigger than this scale. So I'll conclude here my talk. I'll conclude here. Like I mentioned, biologics and biosimilars are more complex than small molecule, and developing biosimilar or biologics are more challenging than small molecule with respect to technical know-how or the technology, a platform, the market demand, market competition, and regulatory challenges. So many steps are there for the biosimilar development. But modern cell line engineering and tools may allow improved title and quality of a biological product. If you want to compete in the market, so we can implement modern cell line engineering tools, which can give you better expression, better quality of the molecules. Robust and consistency processes are the key step in achieving improved title and quality. So we have to develop robust and consistent. Consistency is very, very important to meet the quality of the product. Every cycle, every batch, whenever we develop, we have to make sure that our process and quality is consistent. And that may affect the cost of the development. And last but not the least, biosimilar development offers great opportunity for Indian players. As I stated, a lot of opportunities are there with respect to market, to human welfare, volume, everything. So we have a great opportunity for Indian players. With this, I'll stop my talk. This is my team. I think more than 50 people are there, but this is little older picture. So this is my team, and they work on the biosimilar development, all functions, and we are able to produce 5 molecules now, which are at different stages of development, like clinical, and very soon, maybe next year, we will be launching the product. So my email ID is given here. If you want to approach me anytime for anything, any query or any help, I would be happy to help you all. Thank you very much. If you have any question, please, I would be happy to take your question.
Thank you, sir, for such informative session. Well, I don't see any questions appearing in the box right now.
Yeah.
So I guess
Okay.
participants, if they have any questions, then they will mail you on the mail ID that you had displayed. Thank you, sir, for your enlightening talk.
Okay.
It has
Thank you
helped
very
us in
much.
understanding. It has helped us in understanding the challenges and in biosimilar development, in monoclonal antibodies development, and how to deal with them. Thank you, sir. I now
Thank you very
request
much.
Neha ma'am to address for validatory.
A very good afternoon and a warm welcome to all of you to the valedictory session of RUSA-sponsored national webinar on biosimilars. Like all good things come to an end, we have come to the end of today's webinar. In today's webinar, we had a deep insights into several concepts related to biosimilars. We focused on the concept of biosimilars, its production and characterization, the regulatory authorities and regulatory agencies related to biosimilars. While expressing my vote of thanks, I would first like to thank all the four speakers of today's webinar: Dr. Dilip Pawar, Dr. Jayachandran Ramalingam, Dr. Sanjeev Gupta, and Dr. A. Ramakrishnan for accepting our invitation and sparing their valuable time from the busy schedules. We are very much grateful to all of you. I would also like to thank Dr. Gurunath Pandit, Joint Secretary, Chikitsak Samuha, and other office members of Chikitsak Samuha, Chief Education Officer of Patkar-Varde College, Dr. Mala Kharkar, Principal, Dr. Shrikant Savant, Chief Coordinator, Mrs. Ruchita Dave, for your constant support and for giving us the opportunity to organize this webinar. I would also like to thank the staff members of the Department of Microbiology and Biotechnology for their dedicated work and support in organizing this webinar. Most importantly, a special thanks to all the participants, without whom this event would have not been completed. Your constant support has always encouraged us to organize such webinars on regular basis. Feedback link for today's webinar has been posted in the chat box and it will be active only for 15 minutes. Certificates will be dispersed within a week's time. Thank you all. Stay safe, stay healthy.